Well, good morning, everybody. Thank you so much for sticking with us for a week of virtual OCB. May and I have been looking at the participation numbers, the unique number of participants throughout the week, and I will share those at the end of today's activities. We at least will have Monday through Thursday numbers, and I've just been flabbergasted by the level of sustained participation in the workshop. It's why we're here: science, connection, and community. So kudos to all of you for sticking with us. I hope you've had some fun with your satellite gatherings around the world. And I'd love to see pictures if you post them on Blue Sky or share them with us. We'd love to see what you all are doing, what you're talking about, what things you're nerding out on together. I hope this week has given you the opportunity to immerse yourself in the science that is the reason we're all here and the thing that keeps us going, as well as the connections that we're making, even if informally in the chat. I've seen some really nice connections and just good engagement in general. So thank you for sticking it out. For those who are joining new today, this OCB is really a collective of scientists working at the interface of climate, biogeochemistry, and marine life. Our main science focus areas are listed here in these green icons, and you can find more specific research questions that OCB is interested in on our website. You can find us at all of these places down here. We have a monthly e-newsletter called the Ocean Carbon Exchange. The website is really the most comprehensive source of information about OCB, which has been around for nearly 20 years, and you can find content from meetings we held way back at the beginning in 2006, 2007. So I really recommend that you go spend a little time. There's a lot of information there, and there are a lot of community support resources. We have an email address that will come to all three of us, the three of us, May, Mary, and Heather, here in the project office. Our project office has sustained support from NSF and NASA for the last nearly 20 years, and Woods Hole Oceanographic Institution has been our institutional home. Give us a follow on social media if you're on Blue Sky. Most of our activities are hybrid, so you can find all of the recordings on YouTube. We try to broaden our reach as much as possible because you can only fit so many people in a meeting room. And you can only engage so many people in one time zone, or in a few time zones at one time, as you've probably learned throughout this week.
Thanks so much, Heather and May, for hosting such a wonderful workshop this week. And I am really excited about our last session. It's called Rapidly Changing Systems. So my name is Kristen Krumhardt, and I'm the co-chairs for this session, along with Melissa Melendez and Rachel Stanley. So our session is really focusing on marine ecosystems that are already showing dramatic changes, whether that be from climate change or ocean acidification or other anthropogenic impacts. And so here we're trying to span a lot of things. So spanning a variety of ecosystems from polar regions to the tropics, as well as chemical and biological scales. So phytoplankton, zooplankton, corals, and all parts of the ecosystem. So we're really trying to have that wide view, as well as a wide view of perspectives and timescales. So going from like a long time scale paleo perspective to archived DNA, looking at changes in that over time, or an observation series, all the way to indigenous knowledge. And with this artwork that I made in the background, I'm just aiming to communicate the breadth of all these changes and the impacts that they're having in all different kinds of ocean ecosystems. So yeah, for the just going over a quick overview of our agenda today. So in the first part of the session, we'll start off in the Arctic with Emelia Chamberlain, who's going to be talking about fine-scale biogeochemistry with rapid sea ice melt in the Arctic. And we'll follow that going to the Northwest US or Northeast US shelf, or oops, that's a typo. Sorry. The Northeast US shelf, and Atlantic, and then or Northwest. Yeah, no, it is yeah. Sorry, Northwest Atlantic and the Antarctic Peninsula as well. And then we'll have two lightning talks by Maya Thomas and Lord Coronado Álvarez, followed by a short 5-minute break. And then we'll go to the tropics, where Keisha Barr will be talking about coral reef declines and resilience there. And then each of these talks, they have about 30 minutes each. So the speakers will still have some time for questions. They'll speak for about 20 to 25 minutes. And this will be followed by a panel discussion. So the first one being with Emelia, Tricia, and Keisha. So please be thinking of questions for that. And then for the second part of the session, so this will be after the long break and after the NOAA agency update. So then we'll go to the California with Zach Gold, who will be talking about climate impacts on California current zooplankton, and he's using archived DNA to look at those. And then we'll have Jerry Ivanhoff, who is an indigenous elder from Unalakleet, Alaska, talking about the Bering Sea ecosystem crash. Quick 5-minute break, and then Richard Zeeby, who will be talking about giving us that long time scale perspective on changes during the Paleocene Eocene thermal maximum. And again, that will be followed by a short panel discussion with Zach, Jerry, and Richard. And so overall, one of the guiding questions of our session has been how can all these diverse knowledge systems and timescales inform our response to marine ecosystem transformation? So that's kind of one of the overarching questions of our session. So please be thinking about that. And yeah, with that, I'll just we can start the speakers. And Rachel, why don't you take care of introducing Emelia then?
Hey, thanks everybody for being here, and Kristen for that amazing introduction, and especially the artwork, which is Kristen. So if you don't know, Kristen made all that artwork herself, which I'm just such an awe of. So today I'm excited to introduce our speakers. I'll be moderating in the morning, and then Melissa will take over in the afternoon. And our first speaker is Emelia Chamberlain from WHOI, who will be talking about fine-scale biogeochemical implications of rapid Arctic sea ice melt at the air-sea interface. Take it away, Emelia.
Thank you so much for inviting me to talk in this session. I'm really excited to see how everything kind of loops back together as we think about all these different ways that our planet is changing and how that's changing the ecosystems that depend upon it. So I'm going to start off today talking about ice, which is its own biome. And as a biome, it's actually quite large, all things considered. At any given point in time, it covers about 12% of our oceans. So that's actually quite a lot of ground to cover. Here is from the 4th, so 2 days ago, current sea ice extent for both polar regions. And as you can see, it covers a lot of area from both the Central Arctic, where I'll be focusing on. As a biome, it hosts a suite of organisms from, our polar bears all the way down to our microbes, which is what I'll be talking about today. And so the microorganisms living within sea ice are primary food source for many different organisms within the Arctic food web and contribute to a lot of biogeochemical cycles. And ice algae alone are estimated to account for approximately anywhere from 2 to 24% of total primary production in polar seas. And they're large and in charge. But actually, this estimate is probably an underestimate because most studies only capture the ecology of very flat bottom ice. So all of these numbers are mostly cored from very similar ice types where we have this really flat sea ice that spreads out. We get this diatom-based community on the bottom of the ice, and that's where most of our studies are from. However, ice is pretty dynamic and it's definitely not uniform. So if you spend any amount of time up there, you see that there's many different types of ice, and we're starting to call them microhabitats, where you get these really small scale heterogeneity across different ice. So this is largely dependent on season. In the summer, you'll get these melt features from melt ponds to under ice meltwater layers. Within those melt features, you can get these aggregates of algae, which ASME 2013 has showed can support high levels of biological activity on the scale of the individual aggregate. So this is supporting its own little krill and copepod ecosystem just from these really small areas. You can get these huge patches of the diatom Melissaria arcticus that likes to hang on the bottom of sea ice, and then when you get melt or flooding, they actually detach and sink to the benthos, and there's lots of benthic pelagic coupling through that system. And then you also have different ice formations. So you have ridges. As two ice flows crash together, they form these ridges, and then you get these really protected voids between the different ice pieces where you have, I was just actually on a cruise studying these. I got back yesterday, and we started calling them gardens. So these are our algae gardens growing on the ledges of ridges. So this photo is actually stitched together from a blue eye from about 2 weeks ago. So that was in May, and you already see there's quite a lot of color on these ridges. And then platelet ice is another big one, which has shown to really accumulate algae as it's growing. So there's all these really distinct, unique habitats that have distinct, unique community compositions. And they're experiencing rapid change all the time, from dramatic seasonal transitions and the dynamic spatial shifts that really drive the formation and dissolution of these ephemeral and unique habitats. So here you can see, I think this is about a 2-week span of freeze-up processes where we lose all of our melt ponds. This is a schematic of ridge formation, and obviously we have huge light transitions from the polar day to the polar night. And then they're also experiencing rapid change across the climatic scale, where we're having dramatic losses in sea ice, both in ice extent. So here's the NASA climate change ice extent from the 1970s to about now, where we're losing a lot of our minimum sea ice area. And we also see this kind of modal shift to thinner sea ice. So we're having less multi-year, thick, very ridged ice, and more of this thinner, flat ice that is very easy to move around and drifts quite quickly. So all of these changes are contributing to changes in the ecosystem, but it's actually quite hard to predict how things are changing. And this is from the 2019 IPCC special report, which said that biodiversity projections are limited to key uncertainties regarding the potential for organisms to adapt to change. So we don't really know how they're adapting to their current habitats, so how could we predict changes? And this is particularly true for things like the carbon cycle, where our estimates for net carbon drawdown in Arctic sea ice are really uncertain because we don't have a good handle on those numbers as is. So if we're only looking at productivity measurements from flat bottom ice, we're really not capturing that diversity of community structure that's embedded within these ephemeral microhabitats. And my question to you guys today is, do we think we should actually care about these microhabitats, and do they have an impact on sea ice biogeochemistry? So to study these habitats, you really need to be on the ice for extended periods of time. So this includes drift work. And we were lucky enough that during the 2019-2020 Mosaic International Arctic Drift Expedition, we drifted with sea ice for almost an entire year and captured a lot of that seasonal changes within the ice. And within that, we actually captured a really unique ephemeral environment, which I'm going to kind of deep dive into today. So if you have Arctic summer, there's lots of sunlight, lots of melt. The sea ice and the snow melts can go several different places, but mostly it goes into the ocean. And as it accumulates, it's much less dense than the underlying seawater. And so you actually get this phenomenon where there's not a lot of mixing. You get these really strongly stratified freshwater layers that kind of pool underneath and between the ice flows. And so we actually witnessed these melt layers forming. And when I say fresh, it's really fresh. It's anywhere between 5 to 15 grams per kilogram. And then within a meter to 20 centimeters, you're going to have 32 salinity seawater again. Now, this isn't a new phenomenon. It's actually been documented since the late 1800s during the Fromm expedition, where they drifted, but actually in a sailboat. And you can see in their original diagrams, there's depictions of this meltwater stratification and melt pond flow through. But, you know, since the 1890s, there's only been 9 other scholarly mentions of this ephemeral habitat. So it's not very well documented. And that's largely due to the fact that when we actually go out to sample these environments, usually we're on a giant ship and using a CTD. And if this stratification is only in the upper 1 meter of the ocean, you're not going to capture it based on our traditional methods. But when you do capture it, it's pretty amazing. This stratification really has a huge impact on how biomass is distributed within the upper ocean. And this is particularly prevalent for these open water leads where we would expect to have direct air-sea gas exchange. But when you get this meltwater lens, you have this surface layer of completely fresh water. You have the underlying seawater. And when in the peak of the melt season in July, we actually saw this about 1 centimeter thick just layer of biomass. And that's, you know, going to dramatically change not only who's there, but also what they're doing and what the biogeochemistry is for this air-sea interface. And so that was witnessed in, as I said, peak summer in July, but actually these meltwater lenses were quite persistent spatially as well. So in later summer, in August to September, we actually saw meltwater stratification again in open water leans surrounding a different ice flow. And within that layer, we did not see that 1 centimeter biomass. It was later in the season, a lot less light, but we did see lots of algal aggregates that had kind of accumulated and were buoyant within that layer. So I was very interested in this and wanted to categorize this meltwater microhabitat as part of my PhD. So thinking about the questions, how do these steep salinity gradients really shape sea ice microbial habitats across spatial scales, and what are the potential impacts of meltwater layers to productivity and biogeochemical cycling at this air-sea interface? So to sample these layers, we did the good old duct tape a tube to a stick method and tried to very gently suck up just that surface layer, that interface layer, and then the seawater underneath. And we also did take some samples of the ice surrounding it as well to compare. And we then filled as many bottles as we could to really get a wide suite of biological and biogeochemical parameters taken from these different layers in the upper ocean. And so this ranged from cell counts and chlorophyll to sometimes we got nutrients, but what I'm going to focus on today is actually community composition using DNA sequencing. Specifically, I'll be looking at amplicon sequences, so our bacterial 16S amplicon sequence variants and our 18S eukaryotic ASVs. And then I'll also be looking at productivity measurements from oxygen-argon ratios, where this gives us the biological consumption or production of oxygen to compare for net community production. And so getting into some of the data, this is an NMDS ordination looking at sample similarity within the microbial communities. So on the left is our 16S prokaryotes, and on the right is our eukaryotes. And the sample spread shows that there are really distinct clusters, specifically the meltwater in this bright blue samples were very different from the underlying seawater, not super surprisingly. The interface samples were somewhere in between. Sometimes they were more like the seawater, sometimes more like the meltwater. That might be due to sampling, but occasionally we did get a really strong, unique interface community as well. The other thing that was interesting was the meltwater seems to be closer to the sea ice communities where the meltwater is ostensibly coming from, but they are still unique to themselves. And you can see this down to the ASV level as well. So looking at a heat map of some of the most abundant sequences within our samples. This specifically is our bacterial and archaeal sequences. You can see that we really get these two distinct clusters between those that are seawater samples in dark blue and those that are meltwater samples in light blue. And again, there's more ASV shared between the sea ice and the meltwater. So this is likely a ice-derived community that then adjusts because it's going from very salty ice brines to all of a sudden fresh water. So we're losing a lot of the cells in this process. And when you look at the cell counts, we do see that in both instances, so from both July and late summer, we see the least amount of cells in our meltwater. But what's interesting is that the cells that we do see actually, we estimated using codon usage bias to get an idea of what the growth rates for these sequences might be. And they have much lower doubling times. So the cells that are surviving are very fast growers. They mostly belong to flavobacteria, and they're probably fast-growing aggregate associate bacteria with this algal particles that are kind of trapped in this meltwater lens. We see similar patterns in our eukaryotic communities, so our ice algae and our protists, where we really have these two distinct clusters of taxa, where we have a distinct seawater community and a distinct meltwater community. Again, the meltwater community shared more ASVs with our sea ice, so it's this ice-derived community. But what was interesting is we see that between the peak summer meltwater, so the July, and then the August and September meltwater, these communities are distinct even within this meltwater layer. And when you look at the chlorophyll, that makes a lot of sense because it's later in the season, it's post-bloom, and there's a lot less just chlorophyll. So I think the range in micrograms per liter is up to 0.5, whereas in the July period, it was all the way up to 6 or 7. And what you see here is this really distinct peak in chlorophyll at the interface, where we have this strong biomass gradient that we saw. So we were actually able to capture it. And when you look at this interface community, it's mostly driven by high abundances of pelagophyte ASV. And so the question is this actually growth, or is it just dead accumulated biomass that's melting out of the ice and getting trapped at the salinity gradient? And so we do actually have one C14 derived net primary production measurement from this sample, and it was at about 0.2 micromoles of carbon per liter per day uptake. So not crazy high, especially when you compare it to bottom ice, but there is still some indicators of growth. And then if you look at just add up all of the ASVs from our eukaryotes across these different sample types, you can see that they're really, this yellow is the pelagophytes, and they really take over in this interface layer. So there's some in the meltwater, so they're probably coming from the ice and then thriving at this interface, and we really don't see it much in the seawater below. And when there's been actually quite a few studies on these taxa that show that they have very high salinity tolerance mechanisms. So they're really adept at producing osmolytes and other salinity protectants. So they're probably, again, thriving at this low salinity gradient compared to our diatoms, which did not do as well. And we did not see very many of them alive in this meltwater layer. Now, contrary to our happy growing layer that we saw in July, in our late summer meltwater, we saw a significant reduction of oxygen. So there was a lot more oxygen drawdown in the meltwater layer compared to the underlying seawater. And if you look at some of the nutrient concentrations from these numbers are a bit rough, so not anywhere near the amazing talks we saw yesterday, but potentially indicative of some remineralization processes, because we see accumulate of nitrogen compounds in this meltwater layer. So it could be coming from the ice, but it could be active remineralization. And so the question here is this reduced or increased oxygen consumption signal actually due to active heterotrophy, or is it just reduced production as we have cell death and we're losing a lot of our community? So to test this, I actually brought some cultures into the lab where I took forced melt conditions that were grown up at 1 degree in our lab at UCSD. And so we had 3 distinct ice algal mixed culture communities. There was one that was a diatom isolate from the family Atea and its associated bacterial communities. There was one that was a mixed diatom-based community. And there was one that was a mixed flagellate-based community. And so I grew these cultures up and then put them at 3 different salinity treatments kind of mimicking these 3 layers, what we saw in situ. So there was a 5, a 15, and a 32 salinity treatment, and I let them acclimate over 8 days. And so in this graph, we're tracking chlorophyll across our 8-day acclimation period, and the 2 diatom-based cultures had almost no growth in our lowest salinity treatment, whereas the flagellate-dominated mix community, there is a little bit of chlorophyll production, even in that lowest salinity treatment. However, it is lower overall. That's the arrow pointing at it. So after this 8-day acclimation, I then measured oxygen concentration again over a 48-hour incubation period where I had these BOD bottles, and I tracked oxygen in both the light and the dark so we can get an idea at net community production versus respiration processes. And in this lowest salinity treatment, we did still see net oxygen consumption. So there was part of the community that remained active, even in these algal mixes that did not do very well in our low salinity treatment. So there was this salinity-driven community shift to net heterotrophy, and it was still metabolically active. So it's likely that in situ, they're still quite active as well. And even over just this 8-day acclimation period in our mixed community cultures, we did actually see shifts in the community structure, so shifts in who was present. And in our specifically our diatom-based community culture, we did see a shift away from these pennate diatoms to dinoflagellate species that then started to become more abundant after just the 8 days. So, and in our bacterial community, we did actually see a shift to some more remineralizers as well, associated with nitrogen. So I think that we can safely say there was some salinity-driven community shift to a more net heterotrophic remineralization-based community. And so that's likely what's happening in the field as well. And so this meltwater layer really represents a distinctive but still metabolically active microbial community. And it's going to drastically change the seawater habitat that's at this air-sea interface. These communities are likely seeded from the ice, and then they adapt, showing their resiliency to changing conditions over very quick timescales. However, there is a biogeochemical shift from what was considered a net productive system in the sea ice to a net heterotrophic system at this air-sea interface, which could potentially impact other biogeochemical cycling, from air-sea gas exchange to aerosol formation. When you get these clumps of algae kind of trapped near the surface, you tend to see more, for example, ice nucleating particles. So you're really adjusting a lot of the exchange processes that are happening at this air-sea interface. And as far as, you know, our larger scale changes that are happening, so that's what's happening at the small scale within the ice itself. And then what's going to happen as the Arctic itself continues to change, and we have this reduction in sea ice, and we have thinner sea ice, and we have sea ice that's drifting more quickly, leading to potentially a more dynamic ice scape where we could maybe have more of these open water leads forming. But we could also have them closing more quickly and more mixing. So I don't really know where this is going to go. So that's, I think, an interesting question. We know that a lot of meltwater dynamics are driven by the ice topography itself. So actually, here's a great paper from Daiki Nomura in 2024, which really showed the thickness of this meltwater layer was driven almost exclusively by just thickness of the lead. So the amount of melts was the amount of melts, and then however big the lead was how thick this layer was. And we did see that it actually, at some point, was just completely vertically mixed. And then once we got to freeze processes in the late summer, so I think around September 4th or September 7th was when we really started to see a shift to freezing. And we really lost this layer as it all turned into the new ice and bringing all of those beautiful algal aggregates with it. So I think that predicting future changes is going to be hard unless we do a bit more upscaling and further study on these formation mechanisms for this microhabitat. But hopefully I've convinced you that we do need to consider more than just the bottom ice, and that these microhabitats are very interesting from a biogeochemical perspective. Yeah, here's the satellite images of Lourdes. And then thank you to everyone who made this possible. So a lot of this work was funded by NSF, specifically a grant to my PhD advisor, Jeff Bowman, and the Bowman Lab, all listed here, really helped made those lab experiments possible. So thank you, as well as to all of my co-authors on this work and the Mosaic Expedition field team who kept bringing me samples to filter, even though they had many other things to do.
Thank you so much, Emelia, for that fascinating talk. We have some time for questions, so if anybody wants to raise their hand and ask them, you can also put them in the chat, but while we have this time, if you're willing to ask them, it can make it more interactive. We can start off. Norm has a question in the chat. And Norm said, did you look at meltwater communities in ponds on top of the ice, no connection to the ocean, as well as the leads?
Yes, so we did have a couple, I did look at that as well, and those communities are also distinct. So they are more similar for the bacteria actually to what we saw in the meltwater layers. A lot of those taxa are very similar, and a couple of them, if you're familiar with meltwater taxa, kind of jump out. But a lot of what we saw in the melt ponds was actually driven by sediments that were in the ice. So when this specific ice flow that we were sampling formed, it formed on the Siberian coast and picked up a lot of just like benthic sediments along with the ice. So when we got to melt out, there was, you know, we saw sea stars and leaves and things kind of appearing in the ice. And so a lot of the melt pond communities were associated with those sediments.
Tatiana, do you want to ask your question? You can unmute.
Sure. Thank you for such an interesting presentation. It had so many different pieces, parts. I feel like I need to go back and look at the slides in my own time. You showed us so much interesting data. One question I have for you is, you made some recommendations at the end, and I'm sure I'm not the only oceanographer in the group that's like, we like steady state. We like large areas that are all doing the same thing when we think about like estimates of what's going on. Mm-hmm. And this is such a heterogeneous environment. Have you and your group talked at all about how do we incorporate that heterogeneity? Like, how would we follow those recommendations, or what are some promising routes forward to incorporate the heterogeneity?
Yeah, and so that's actually, I think that's the bane of sampling sea ice is, one ice core next to another doesn't guarantee that they're necessarily the same. So I think right now, what we need to do is actually just start compiling what's behaving similarly. So when do we see similarities in community composition or rates? And maybe from there, we can start building either mechanistic, or even if we get enough data, maybe go the machine learning route and kind of start to pool together these similar community types that we could then maybe adjust into predictive models or things like that. Because yeah, we want our steady state. The reality is it's very not steady, but likely on larger timescales with enough observations, we can start to pull out patterns that are steady state.
Thank you so much.
We have another question in the chat from Suzanne saying, did you look at hypersaline brine channels?
So I actually, not on this cruise, but I'm very excited because the cruise I was just on, I did actually collect some only brine as well as melted ice, as well as ponding and things like that. So I haven't been able to make those direct comparisons, but I know that the brine channel communities should be similar to what we're seeing in the melted ice cores, because we melted them with a buffer. So hopefully most of them were retained. So that would be most similar to the ice samples that I showed.
I have a question. I'm really fascinated by the possibility for the algae to block air-sea gas exchange. Was anybody doing air-sea sort of measurements, flux measurements on the cruise that noticed if there was a measurable effect of that?
Yeah, so they put a chamber on the water when there was the melt layer and the flux was 0. So it was blocking it just as effectively as the ice. So it was interesting because we were kind of expecting like, oh, the lead's open, it's not refreezing, there's going to be all this exchange. And then they put the little chamber out and it was literally 0. So that was really interesting to see. And I think there's a lot more like studies moving forward that people want to do and thinking about that. They're also, from a particle perspective, like aerosol perspective, contradictory. The people who were measuring INPs saw an increase in biological INPs from melt ponds. So I think it's, maybe you might know more on parsing through some of those dynamics, but there were some interesting air-sea gas exchange puzzles that we encountered due to this.
That's so cool. Well, thank you so much, Emelia. And if you continue to have questions, you can put them in the chat. And then also we have a panel later this morning where Emelia and our other morning speakers will be available to answer your questions. So next up, we're going to be hearing from Tricia Thibodeau from the University of New England, who is going to be talking about riding a wave of variability through plankton time series from the Southern Ocean to the Northwest Atlantic.
Thank you again, Rachel, for the introduction, and thank you all for tuning in this morning. I wanted to start my talk today with the end, or the main point, which is that high degrees of variation in time series around the globe highlight the need for long-term monitoring to improve signal-to-noise ratios and identify trends in these highly variable environments. And this conclusion was originally developed based on my research analyzing phytoplankton biomass data across various time series. And so this figure has the coefficient of variation as a percent on the x-axis, and then different time series data sets on the y-axis, all in the northern hemisphere. But what you can tell is that whether you were in an estuary like Narragansett Bay at the top, or in the open ocean gyre like the Sargasso Sea, there was a lot of variation among these different data sets. And so having long-term time series helps us to elucidate the long-term trends in these areas. So today I want to emphasize my main point further by taking you on a tale of 2 time series. These are time series that I had the pleasure to be involved with during my career as a graduate student and postdoc, and now as an assistant professor. And I say 2 or 10 time series because while I'm only going to focus on 2 distinct data sets, the Palmer Antarctica Long-Term Ecological Research Program, or PAL-LTER, and the Northeast U.S. Shelf LTER data sets, the trends and conclusions I present are really based on results from multiple time series data sets in this region. And so the first time series I'll focus on is located along the Western Antarctic Peninsula, or what I'll refer to as the WHOI, and it's located across from South America. And it's been experiencing long-term temperature warming. This figure shows winter air temperature on the y-axis and then year on the x-axis. And we've seen a long-term temperature increase with an average winter temperature of 1.1 degrees centigrade per decade over time. We've also seen a long-term decline in sea ice in this region. This is sea ice duration in days on the y-axis and then year on the x-axis. And overall, we've seen a long-term decline in sea ice duration, such that we've seen an overall decrease in 3 months of sea ice season since 1979. In the Northeast Shelf region, which is located again in the Northwest Atlantic here, we've also seen long-term warming. This is temperature on the y-axis and then year on the x-axis. And overall, we've seen a mean annual water temperature increase of around 2 degrees centigrade over the last 150 years. And while this rate isn't quite as rapid as those that we've seen along the WHOI, I'll draw your attention to the gray bar that we see here, in which we have seen particularly rapid rates of warming in the last decade, especially in the Gulf of Maine. And while these rapid rates of warming may be alarming, thankfully we've had these two time series data sets, the PAL-LTER and the NES-LTER, that have characterized and documented these long-term changes and helped us to understand how they're affecting the food webs and ecosystems in both of these regions. And so I'll first take you on a tale of the PAL-LTER, which, as I mentioned, is located along the Western Antarctic Peninsula. And every year since 1993, scientists have gone down in January, which is the austral summer, and covered about an area the size of Oklahoma in 30 days. And they've traversed along these series of grid stations that you've seen here on these bright orange ships, so the Lawrence and Gould and now the Palmer on the bottom right. And they've sampled everything from the bacteria and the plankton up to the penguins and whales. And we're really interested in characterizing this north to south climate gradient that's developed, in which the northern part of the peninsula is now considered this subpolar climate and no longer has perennial or year-round sea ice. And it's really this warmer, moister climate than it used to have. Whereas in the far south region of the peninsula, we still have a truly polar climate that's cold and dry. And so the PAL-LTER is interested in understanding how this climate gradient is impacting the ecosystem over time. And so I'll go through some of the long-term changes that have been observed over the last 35 years. To do this, I'll show you a series of standardized anomalies plots in which we have the long-term standardized anomalies in the dotted line. We have the smoothed multi-year variability in the unbroken line, and then significant long-term trends in the dashed linear lines. And as you can see, and as I mentioned, we've seen a long-term increase in seawater temperature and a long-term decline in sea ice. I'll note that there's been a lot of variability in the sea ice over the sampling period, in which we generally saw a long-term decline in sea ice until about 2010, when we actually started to see an increase in sea ice until about 2015. And then it's taken a turn since then, and we've now seen some of the lowest recorded sea ice concentrations in the data set's history. And a lot of these changes are tied to shifts in climate oscillations, especially the El Nino Southern Oscillation in blue, and then the Southern Annual Mode in black. And as you can tell, the SAM has actually increased in the number of positive anomalies over time, and this affects the way the wind moves across the sea ice and ultimately can decrease the amount of sea ice that's being accumulated in the region. In terms of food web impacts, we've also seen an increased amount of storminess over time, which then impacts the mixing of phytoplankton, because if there's less sea ice and more wind moving over the water column, phytoplankton are typically mixed below their critical depth and can't photosynthesize. And we see for phytoplankton that there's been a general stabilization of phytoplankton biomass over time, with a recent uptick in positive anomalies in the last decade, although we do see distinct sub-regional differences in which the northern part of the WAP near the Palmer Station has seen a long-term decline in phytoplankton biomass overall and a shift in community towards smaller flagellated cryptophytes shown here. Whereas in the southern part of the WAP, we've actually seen an increase in phytoplankton biomass over time, dominated primarily by large, fast-growing diatoms, because as the sea ice that was once permanent there has now begun to decrease, it's released phytoplankton from light limitation and allowed them to grow more than they used to be able to. In terms of the zooplankton, we've seen variable trends overall. For the Antarctic krill, shown here on the bottom left, its abundances along the WAP have relatively remained stable, which is somewhat surprising since we know that sea ice has been declining over time and this is an ice obligate species. But we've actually been seeing a contraction of the overall krill population throughout the Southern Ocean. And so in areas north of the WAP where the waters have warmed more and sea ice has been lost at a more significant amount, we've seen those krill populations move south into the WAP region, and so this has helped to stabilize the population over time. In terms of whale populations, especially humpback whales, shown in this beautiful bubble feeding image down on the right, we've seen oscillation in their abundance with a recent decline. Although when we compare that to the long-term trend, when we think about whaling and whatnot that had impacted and decimated those populations, their abundances really have rebounded quite well recently, especially as sea ice has continued to decline, increasing their foraging grounds in the area. So I couldn't help but do a quick shout out to my pteropods, my favorite zooplankton. This is a video of them swimming, and as you can tell, they're commonly called sea butterflies. I was excited when May told me that this video is still getting hits on the OCB website, on their YouTube page. But I wanted to mention them also because pteropods are considered a gelatinous zooplankton, just like the pelagic tunicate, the salps. And overall, we've been seeing an oscillation in their abundance over time, but no long-term directional trend for Limacina in this case, but then others we've actually seen increases like for the salps. And this is mainly because these gelatinous zooplankton are considered ice obligate species like the Antarctic krill. And so this figure here shows the abundance anomaly for Limacina in the northern part of the region, and then for the south and far south regions of the WAP. And I've highlighted the most recent year in 2023, in which we saw some of the most positive anomalies for Limacina. And this was a particularly low sea ice year along the peninsula. And so this figure shows sea ice anomaly on the x-axis and Limacina abundance anomaly on the y-axis. And you can see that when there are higher number of sea ice days or a larger ice area, that typically corresponds to lower Limacina abundances. And we see similar relationships, as I mentioned, for the salps. And so overall, these results indicate a polar ecosystem along the WAP in transition, which 35 years ago, when the PAL LTER first started sampling, we typically saw a system shown here where there was high sea ice, and there was a short linear food web that went from diatoms to krill to higher trophic level organisms, and generally high amounts of carbon export. Whereas now we're seeing more of this subpolar system where there's lower sea ice and increased storminess, which deepens the mix layer. And then this leads to long, more complex food webs where we see smaller flagellated phytoplankton dominating the base of the food web. These are then grazed by krill as well as gelatinous zooplankton, and then these in turn are eaten by higher trophic level organisms. But we overall see a general decline or a decrease in the amount of carbon export. And I encourage you to stay tuned for the talk after mine, which is by Maya Thomas, and she'll be talking more about the effects of zooplankton on carbon export in the WAP. And there's still an unopened or an unanswered question as to what the pace of this transition from this short linear food web to this long complex food web is going to be. And so this necessitates the need for continued monitoring so that we can better understand the abundances and sustain these different populations that are so important to this polar ecosystem. All right, so now moving to the northern hemisphere along the northeast U.S. shelf or in the northwest Atlantic. And when I talk about the NES, I'm really talking about two distinct subregions. There's the Gulf of Maine, which has its own distinct circulation in which there are currents that come from the north that enter the Northeast Channel. And there's also currents that stem from the Gulf Stream that enter the Northeast Channel. And I'll talk about more about how that affects the Gulf of Maine in a moment. We then have the Mid-Atlantic Bight region, which is the area that the NES LTER has been working to characterize. And for almost a decade now, this group goes out every few months to look at seasonal differences along the shelf break in terms of the physical environment as well as the food web. And we, as I mentioned before, have seen long-term warming in both of these subregions of the NES. These are sea surface temperature anomalies on the y-axis and then year on the x-axis. And we've seen long-term warming in both of these regions, especially in the last decade, as indicated by these dark orange lines here. We've also seen circulation shifts in these regions. And so this figure here shows slope water proportion as a percent on the y-axis and then year on the x-axis. And I mentioned earlier these different slope waters entering the Gulf of Maine. So the Labrador slope water that's typically colder and fresher versus the warm slope water that stems from the Gulf Stream that's warmer and typically lower in nutrients. And we've seen in the last decade that there's been an increased proportion in the amount of warm slope water entering the Gulf of Maine. And a lot of that has to do with a northward migration of the Gulf Stream, helping to move some of that warmer water closer to the Gulf of Maine. And so these changes in the physical environment of the NES have impacted the food web and nutrient cycling, which the NES LTER is working to characterize. And so they've found that differences in seasonality between the Mid-Atlantic Bight and the Gulf of Maine result from an interplay between nutrient and temperature effects. There's also been synchrony among taxa, especially the copepod Calanus finchmarchicus, which varies seasonally and spatially, but that local habitats also play an important role in driving this variability. And we've also seen strong shifts in trophic transfer coupling from high trophic transfer in winter to low in summer. And so this image here kind of documents those two different trophic pathway states, in which in the winter, we typically see this state 1, which is characterized by larger phytoplankton, grazed then primarily by microzooplankton, and then eaten by crustacean zooplankton. Versus in the summer, we typically see the second state of smaller phytoplankton and as well as an increased reliance on the microbial loop, which are then eaten by microzooplankton, as well as some gelatinous zooplankton. But we still had this unanswered question of what the fate of excess primary production was in summer, because we were observing high primary production rates along the NES, but low phytoplankton biomass. And typically we would expect to see this excess biomass grazed by microzooplankton, but we saw low microzooplankton grazing rates. And so to answer this question, we did a cruise along the NES LTER in summer 2022, where we did a series of grazing experiments with microzooplankton as well as macrozooplankton. And we found that salps really dominate the summer grazing signal along the NES. And so this figure here shows the grazing loss per zooplankton biomass on the y-axis and the different macrozooplankton on the x-axis, with salp aggregates being typically the smaller size of the salps versus the salp solitaires being the bigger ones. And the main species of salp that we were seeing were these Thalia democratica, which I grabbed a couple photos of from the cruise here. And overall, we found that salps were removing the largest fraction of phytoplankton carbon when they were present. And when we included macrozooplankton in our grazing terms, this doubled the trophic transfer that was originally accounted for. So this figure shows micrograms of carbon per liter per day on the y-axis, and then primary production in green and grazing loss in orange. And we found that when we only included the microzooplankton grazing term, that accounted for about 30% of the primary production consumed. But when we also added in the macrozooplankton grazing term, that accounted for overall 60% of primary production consumed. And so this helped us to better understand where a lot of that excess phytoplankton biomass was going in summer along the NES. And I wanted to take a moment to talk briefly about some related work I've done focusing on long-term trends in phytoplankton biomass in Narragansett Bay, which is located very close to the Northeast Shelf LTER sampling region. The Narragansett Bay is home to the Narragansett Bay Long-Term Plankton Time Series, which is one of the longest phytoplankton time series in the world, with weekly counts of phytoplankton since the 1960s. And it's located just north of the first NES LTER sampling station near Martha's Vineyard. And what I found by analyzing the 60-year data set was a long-term decline in phytoplankton biomass. And I used the dynamic linear model to interpolate some of the missing data that we had, as well as to analyze the long-term trend. And so this figure here has chlorophyll A on the y-axis and year on the x-axis, and the little gray dots you see are the actual observed data, which you can tell are quite variable. I didn't fully appreciate what variability meant until I started studying an estuary. And then the black line you see here is the seasonal component of the model, and then the green line is the long-term trend, which you can see has declined over time. And we also looked at this by observing the cumulative chlorophyll A by comparing the first decade to the last decade. And overall, we found a significant decline of 49%. And this time series result further emphasizes my point of needing long-term data sets to fully elucidate significant trends, particularly in these highly variable environments like estuaries, as well as the importance of incorporation of local or regional habitats into population analyses, because they also likely play an important role in driving variability affecting ecosystem synchrony. Finally, I wanted to finish by highlighting some recent rapid changes that have occurred along the NES in 2023 and 2024. So in 2023, we saw that summer bottom water temperatures in the Gulf of Maine were the warmest on record since 1959 and represented the second largest bottom water marine heat wave in the area's history. We also saw a widespread bloom of dinoflagellate Tripos muliarii, or seratium, shown here on the left. And it generated a record high chlorophyll concentration since 1999, which was at almost 10 times that of the average in some places. So this figure here shows the chlorophyll anomaly for June 2023 relative to the long-term mean since 1998. And we can see that especially in the Gulf of Maine, but really throughout the NES, there were positive chlorophyll anomalies in this area. And you can just get a sense for the extensiveness of this massive chlorophyll bloom. And we're still figuring out some of the mechanisms that led to this bloom, but it was a very wet year that year, and it was also lower than average PCO2 concentrations. In contrast, in 2024, while it was the warmest year on record globally, the NES actually saw colder than average temperatures in this region, especially in the Gulf of Maine. And so this figure shows the temperature difference comparing the February 2024 sea surface temperatures to that of the long-term average from 2000 to 2020. And you can see that, especially in the Gulf of Maine, this area was colder than average, and there were distinctly colder waters off of the Northeast Shelf break here. And we found that this was mainly due to an increase in the proportion of the Labrador slope water that was entering the Northeast Channel into the Gulf of Maine relative to the amount of warm slope water that was entering the Gulf of Maine stemming from the Gulf Stream. And this was mainly because the Gulf Stream had actually migrated further south than it had in the last decade. And we're still figuring out some of the ecosystem impacts of this bloom or of this temperature change. Interestingly, we didn't see a big Tripos bloom like we did in 2023. And we did see some migrations and changes in the migrations of different fish species in this region. So again, I hope the examples I showed you today from the PAL LTER and the NES LTER help to highlight how high degrees of variation in time series around the globe highlight the need for long-term monitoring to improve signal-to-noise ratios and identify trends in these highly variable environments. We saw that with the PAL LTER along the WAP, there's been a long-term increase in temperature and a long-term decline in sea ice, although this has been highly variable over time. And along the NES LTER, we've seen a long-term increase in temperature and changes in ocean circulation. I think it's important to note that in the last 10 years, we've actually seen some of the most drastic physical changes in this region, in both of these regions, in which sea ice has declined much more rapidly than it has in the previous decades. And we've also seen a much more rapid temperature increase in the NES-LTER region. And these recent, we've also seen that these shifts in the physical environment are impacting the food webs in these regions, in which we're shifting from these linear short food webs to these more complex food chains, with potentially decreases in the resulting carbon export and abilities to affect higher trophic levels in these regions, which impacts management. And these recent rapid changes also highlight the need for continued observations to assess the full ecosystem's effects and to quantify the pace of these changes, especially in these ecosystems in transition. And I also feel like I should add, especially in light of some of the very recent news last week, that NSF plans to not renew the contract for the only Antarctic research vessel that it currently has, the Nathaniel B. Palmer, that we as scientists need to continue emphasizing the importance and need for these continued data sets. And so I hope my presentation today gave you some talking points that you can bring to your representatives and even to your neighbors. And with that, I'd like to thank my funding sources and my collaborators, and a special thank you to OCB. I love OCB, and I'm honored to get to present to you all today. I just wish we were in person, but I really appreciate all the work of the steering committee and this special session on rapid change to make this presentation still happen and this conference still happen. And with that, I will take any questions. Thank you very much.
Thank you so much, Tricia, for that fascinating talk. So again, we have a few minutes for questions. If anybody wants to put them in the chat or raise their hand, that would be great. Well, people are thinking, I had a question. So you mentioned in the Palmer LTR how there was the shift to from the sort of high export system to the sort of more convoluted food web and potentially lower export system. Was that just in the part of the northern part of the peninsula where it's been warming, or was that throughout the entire peninsula?
Yes, typically it's been observed mostly in the northern part of the peninsula, where we do see those lower amounts of sea ice and more mixing. And I think the question the PAL-LT are still trying to figure out is like when and how much that food web might migrate further down into the south and then the far south regions of the peninsula moving forward.
Thank you. There's a question in the chat. Let me see. It's sort of similar, actually, which is, have you observed shifts in the phytoplankton composition between the northern and southern regions over the past decade? If so, how have you investigated these shifts in terms of how they may influence trophic dynamics, particularly in relation to the feeding selectivity of key grazers such as euphausids and salps?
Excellent question. So yes, we have seen changes in the populations in the last 10 years or so. As I mentioned, primarily this shift from cryptophytes in the northern part of the WAP, and then this increased presence of diatoms in the southern part of the WAP. Generally, what we think happens with this shift in the community is that the smaller phytoplankton tend to be grazed more efficiently by the gelatinous zooplankton like salps and pteropods, because they don't get because they tend to get caught in the appendages of the krill when they're trying to use their appendages in their basket feeding to feed. And so they do think that that can affect the grazing efficiency and might be one reason that we could see declines in krill populations. And then, yes, so I think that's kind of the main answer there.
Awesome. Thanks. Suzanne, do you want to ask your question?
Thank you. Thank you, Tricia, for the shout out on behalf of our research fleet. I shared a link there, but it's really important that our community speaks up, even if it seems futile right now, but it is important, and organize with your scientific societies. And I'm hoping you can solve a forever question. You point out that all is really essential variability, and we're always pressed because it's important to describe average states. And I'm calling for our community. I'd love to work with people on pointing out how these small scale interactions are really important, and they don't lend themselves to averages. Do you have examples from your time series where you just have to take a particular state and accept that? So I'm thinking of, for example, a cold or a warm year, that's characteristic rather than some, this is the average temperature in winter. Sorry, that was very lengthy, but I'm hoping we can work on those kinds of questions.
Yeah, no, it's a great point. And maybe in our discussion, we can talk about it more because I feel like Emelia also was sort of,
Yes.
her results sort of hearken to that, your point of like, yeah, this small variability can actually, or these small regions of variability can have important implications. And I think, again, that's where the power of these long-term trends can come from because it helps, or these long-term data sets come from because it helps provide context for when we are seeing maybe these more rapid or uncharacteristic variability in the data set. I do think, we kind of, and I sort of tried to simplify it in this sense with the different food webs, but like you do typically see like this colder, sea ice driven system with the short linear food web. And so typically when we see a high ice year along the WAP, like that's when we would expect to see that kind of system. And at least for the most part, that tends to be the case. And, I think along the NAS, like we also see that and that, you can kind of say the summer tends to be the less efficient food web and the potential gelatinous grazers. So I think we can like have these general ecosystem states, as you said, but then maybe just these like massive error bars around them to help emphasize that they're always subject to change, which is why we need to keep monitoring them.
Yeah, I'll just add that including departures from those averages, like very specific examples of departures, is quite helpful.
Yeah, for sure. So maybe showing the long-term, some of these ecosystem states, but then examples, as you're mentioning, of like areas where they depart to make sure that those are included in the conversation too. And it's not just the stagnant food web that we're observing.
Thank you for a great talk.
Thank you.
Thank you so much, Tricia. So there are some more questions in the chat, but we'll just wait for the panel for those. And we're going to have some wonderful lightning talk.
So yeah, thank you for letting me give this talk, and thank you a lot to Tricia before me for really teeing me up for a lot of stuff. But today I'll be talking about the seasonal and interannual variability in zooplankton carbon export in the West Antarctic Peninsula. So I look at zooplankton and their relation to the biological carbon pump, particularly through their fecal pellets, is what this talk will be on, and the sinking of those fecal pellets. And just like what Tricia was saying in the last talk, we are going to be in the Western Antarctic Peninsula, and instead, throughout the entire peninsula, we're going to be kind of focused at Palmer Station. And all of the work I'll be talking about has been done at Station E, which is this about 5 kilometers from Station. And that's where all of the work I'll be presenting has happened. And yeah, just big shout out to my advisor and my collaborators from WHOI. Cool. So the zooplankton that we see in this area are typically these 3 guys right here. And from left to right, we have krill that create long cylindrical fecal pellets that in this system are actually kind of moderately fast sinking because they only sink about hundreds of meters a day. And then we have copepods that create these small ovoid fecal pellets that sink less than 100 meters a day. And then salps are the kind of big player in this area that have these large tabular carbon-rich fecal pellets that can sink up to thousands of meters a day. And we collect these fecal pellets in these bottom moored sediment traps that we have at Station E. And in these sediment traps, I put a viscous cryogel in the sediment trap so I can individually then image and analyze all the fecal pellets that we get. And I've done that over the past 2 seasons, 2 austral summer seasons at Palmer Station. And this is the data from those 2 seasons. So I went my first season was a 2023-2024 austral summer season. And we got really good temporal coverage throughout the season, and we saw this great decrease in the amount of fecal pellet flux throughout the season. And this past season, we got a little bit less spatial or temporal coverage, but it's very fascinating to see that the magnitude of the flux was very similar between these 2 seasons. And in the next slide, we'll get into what the specifics of what that is looking like. So again, my first season is top and my second season is on bottom. And on these graphs on the left, we were looking at zooplankton biovolume that we collected in plankton nets. And on the right, we're looking at the kind of specifics of the pellets that we saw in the sediment traps. And so we kind of have separated these two seasons by what we're seeing. In the first season, we call it a krill year because the majority of animals we caught in our nets were krill, and in the majority of fecal pellets that we caught in the majority of fecal pellets we caught in our sediment trap was cylindrical fecal pellets. So that kind of match was really awesome to see that the sediment traps are very nicely reflecting the zooplankton community around it. And that kind of changed in this past year. In our second year is what we would consider a SELP year. So there were SELPs everywhere throughout the entire season we were there, and they were the vast majority of animals that we caught in our plankton nets. But it was interesting that our sediment traps didn't exactly reflect that, where we actually saw, we still were dominated by cylindrical fecal pellets. So that was a difference that but between the two seasons that we were looking at. And then if we go to the next slide, we can kind of get into that first season and how well it reflected the trends that we expected to see in the zooplankton community. So previous work from our lab, from Dr. Jack Conroy, found that there is a difference in there's a change in the dominant krill species throughout the season, going from the large dominant Euphausia superba to the much smaller Thysanessa macrura throughout the summer season. And we think that that change in species is a better reflection of the reason we saw this decrease in particularly cylindrical fecal pellets throughout the summer season. Where at the beginning of the season, in this top graph, we can see that we are getting the majority of the krill that we were getting were Euphausia superba, and then there is a shift in the later season to Thysanessa, and that the decrease, and we expect that the decrease in cylindrical pellets that we're seeing in the graph below it is less because of general krill abundance and more because of the change in the species that we are seeing throughout the season. So that reflection was a great reflection to see the zooplankton community seasonal succession also reflected in the sediment traps. I think I'm out of time, but thank you so much, and I'd be happy to take any questions if there's time or at the panel discussion.
Thank you. We don't have time for questions now. You have just about 5 minutes, but feel free to put them in the chat, and maybe Maya can answer them in the chat or at the panel. And next is Lourdes. If you want to introduce yourself and start talking, that would be great.
Hello, everyone. I'm Lourdes Coronado. This research began with a simple question. Can we quantify RC CO2 fluxes during hurricane season using satellite data? We have access. We had the access to satellite database surface PCO2 from the Pisces Nemo model, as well wind, temperature, salinity data from the same period. So using the Starland equation, we estimated FCO2 from May to November 2024 along the tropical Mexican Pacific. And at first, everything looking normal until we're focusing in Hurricane John, which crossed the region in September 2024. There are no visible significant change in the PCO2 or FCO2 time series. That was a huge surprise because hurricane caused a strong vertical mixing and rapid surface change, which usually increase CO2 outgassing. The question is, there was the satellite PCO2 underestimated during the storm. So with satellite data, temperature, wind, and salinity, we correct these PCO2 values using a Bayesian neural network recurrent model. We trained it using data from Hurricane Lydia in 2017 because we had a high frequency time series of PCO2 measurement from a buoy and satellite inputs like temperature, salinity, wind, and chlorophyll. And So when we then applied the training Bayesian neural networking recurrent to correct satellite PCO2 data during the whole hurricane season of 2024 in front of Guerrero State, where 3 storms occurred, John, Ileana, and once E. With the correct PCO2, we recalculated FCO2, and you can observe this comparison between the original and corrected PCO2 and FCO2 in the graphs that you can see in this slide. During hurricane, PCO2 increased quickly, while FCO2 reached a peak up to 35 millimoles of carbon per square meter per day, which are not visible in the original data. This storm contributed about 25% of total FCO2 of whole hurricane season from May and November 2024. So this shows that if we don't correct satellite derivative PCO2, we may miss a significant part of oceanic outgassing during this kind of events that are recurrent every year. Thank you very much.
Thank you so much. And we have time for one question, or anybody has a question? Maybe people need a moment. When lightning talks, it's always hard because the talk itself is so fast. Also, think about it.
Too much information in a little time.
It was fascinating. Do you have plans? Do you know if there's another, was it just happenstance, the buoy that had the hurricane nearby, or was that like intentionally placed in a hurricane strong area? It seems like it would be great to ground truth it with more than one storm if the data exists.
I want to prove this neural networking, Bayesian neural networking in other sites, for example, in the Caribbean, Mexican Caribbean. And I want to train it again if we have good results with other hurricanes in the last year.
And then one question from the chat, what satellite PCO2 product did you use?
We used Copernicus Marine Service products. Is type L4. I have the DOI. If you are interested, I have the DOI and I will type in the chat.
That would be perfect.
Listen, thank you.
Thank you so much. We'll start now after our break with Keisha Barr from Texas A&M, who's going to be talking about rapidly changing systems, coral reefs, climate impacts, bleaching dynamics, and pathways to resilience.
Thank you, Rachel. Yeah, a lot of stuff in one talk. You can see my screen okay, right, Rachel? Okay, perfect. Thank you. So good morning, everyone from Texas. Thank you so much for, Melissa, for you inviting me to give a talk about coral reefs. Anytime I have an opportunity to talk about corals, I love to do that. And I think it's an appropriate talk regarding this session regarding rapid changing systems. So I'm going to talk to you more about what we've observed, some areas that we've seen resilience in, and potentially what we can do about it moving forward. So I'd like to just first start showing a beautiful, healthy coral reef. So coral reefs are built of individual coral colonies, and these coral reefs can provide up to support for up to a quarter of our marine life. But what we're seeing with our coral reefs is that they're degrading over time, where we're seeing that these changes are occurring so rapidly that there can be a change in the corals from 1 year later, we have a beautiful, healthy, thriving coral reef, to a degraded reef in such a short period of time. So this becomes very alarming because when we look about coral reefs across the world, we have them in all areas of our ocean. So this image here, this diagram is showing you where we have corals. All the pink dots are coral reefs. That yellow star in the middle is showing the previous image of the coral reef there. So you can see it's very far from land and has little to no human influence directly. But regardless of where we look in our oceans, we are seeing that there's a decline in coral cover, or the amount of live coral on reefs. So if we look at the Seychelles, we see that there's a decline. We also see that there's a decline at the Great Barrier Reef. A couple years ago, as you guys might have seen the obituary, that the Great Barrier Reef was declared dead. But there's still live coral there, but there has been a decline. Of course, in the Caribbean and Belize, in the larger Caribbean, it's a pretty tragic story regarding the amount of coral cover we have in these areas. So it's currently estimated that we've lost about half of our coral reefs in the last 30 years. And this decline is happening twice as fast as rainforest decline. And current estimates depict that we might have coral reefs completely gone by 2070. So this is a pretty dire situation regarding how fast these systems are changing and what are the ramifications of that change. So as we all may know, the changes that are impacting coral reefs are impacting all of our other systems. We like to characterize these as local and global factors. So local land-based runoff, eutrophication of systems, and also warming and acidification. And when we look at coral reefs, we look at the impacts of these different stressors on the corals and also their response. One of the best ways for us to look at coral response is through coral bleaching, because corals have a symbiotic relationship with a dinoflagellate. And when there is a stress, the corals do undergo bleaching or the loss of that symbiotic relationship. So they visually turn white, which makes our job pretty easy to tell if a coral is healthy or not based on the pigmentation of the coral. So what my lab does is we try to understand that these mechanisms that drive coral reef decline and also the response. Why do we see variability in the response of corals in different areas within species or cross species? What are driving those mechanisms? What are driving the mechanisms of their growth or their calcification, and also the mechanisms of bleaching? And then we also take this full circle and look at the applied angle. So what do we do about it? We see that this decline is happening so fast. We need to have some kind of action. And I think all of us working in any type of climate science, we understand that the only way that we can slow the decline is influence or try to change human behavior. So we're trying to connect what we're learning regarding the mechanisms to providing some solutions, and also engaging with folks to help mitigate some of these impacts that we have on reefs. So I want to talk to you all today about a place near and dear to my heart, Kaneohe Bay, Hawaii, on the east side of Oahu in Hawaii, because I think this is a very interesting system where we have seen change over time, or there has been documentation of change, and also a reverse of that change. So we've seen that this coral reef has gone from a coral-dominated system to an algal-dominated system and then back. And it's one of the only systems that has been documented to be able to recover from an algal-dominated system. So the thing I like to tell the story about Kaneohe Bay is we do know a lot of information about the bay because of the early information from the Hawaiians and Polynesians in this area, where they've documented, or there's oral tradition regarding the overall coral cover of Kaneohe Bay and how that has been relatively high right before Western contact. Once we had Western contact, of course, we had a lot of land use changes associated with that, which also caused a lot of input sedimentation into the bay that has influenced the coral cover and declined. We've seen that decline over time, either documented through oral traditions or also some written documents as well. Also in the bay, there has been, of course, land development and urbanization, but we've also seen extensive dredging for the ship channel and the construction of a naval base there. So I'd like to just show this image of the Hawaii Institute of Marine Biology, where that is located, or Moku'oloi is the island in Kaneohe Bay. And you can see that there were parts of reefs, so this reef up here, or entire reefs such as this reef down here, that have been completely removed. So with this dredging, of course, removal of an entire reef will influence your coral cover, but also we're unsure what they did with that material after they dredged those areas. Some believe that that was deposited right back into the bay and also influenced the coral cover as well. So we saw rapid declines in the 40s and 50s in this area. And if that wasn't enough, for nearly 30 years, there was primary and secondary treated sewage that was directly discharged into the bay. So this is what caused a huge algal bloom in the bay, an increase in algae, and also with that, a smothering of the corals. We saw a huge decline. So this reef ecosystem was declared a system under stress. The local community took action and they got the sewage diverted to offshore waters. And what we saw is once that sewage was diverted, the corals started to come back. So I think this is a really important point, that a lot of the times when we think about decline in our reefs or in our systems, we think that there's nothing we can do about it, or we won't see an immediate action. This is an example of how they did see a pretty fast response to the decline, or to the removal of the sewage. The problem with that is, with these local impacts being reduced or subsiding, we do see that the global impacts are having more of an influence, where we have now had 4 documented bleaching events or warm heat wave events happen in Kaneohe and across the Hawaiian Islands, and that is now threatening the coral cover in the system. So having this long-term view, or this long-term data, which a lot of us have been talking about this week, is so essential for us to understand what impacts are driving these changes, and predict in the future how we might see these systems respond as well. So I just want to briefly talk about warming, because this is the primary factor that we've seen is driving the decline of corals. The amount of warming, there's a threshold in which if it gets too hot for corals, they lose that symbiosis with their algae, they bleach, and then if it stays too hot for too long, they do die. And we've had 4 documented bleaching events. The first was in 1996, so we don't have the sea surface temperature or the coral satellite bleaching alerts for that. But in 2014, 2015, and 2019, we had varying degrees of warming in these areas. So the darker the red that you see here, the more likely we'll see mortality of the corals. And the lighter colors, the lighter yellows or blue, if you see any, would indicate there's little to no stress on the corals. So in 2014 and 2015, this was our 3rd global bleaching event. So all coral reefs in each area of the ocean experienced bleaching. Hawaii got hit twice with that. And then we also had another event in 2019. And what we see is that the warming did have a different degree of different impacts on areas on the north side of the islands in 2014, the south side of the islands in 2015, and then 2019 was kind of a mixture of the two. So what we do to try to understand the impact of warming is we essentially go out and just look at corals. And it is a very subjective process where we're trying to characterize the pigmentation of them. Do they look normal in pigmentation? Are they paling in pigmentation? Are they bleached white, or are they dead, newly dead? And the interesting thing about this is that we can do these surveys from the boat on a nice calm day and hit a lot of different areas in Kaneohe Bay here and try to document where we're seeing high levels of bleaching and high levels of mortality, and try to see what are the drivers of that bleaching and mortality in that area. Or if there is low levels of bleaching, why do we see low levels of bleaching in that area? So in 96, they saw a lot of the bleaching was restricted to the inshore reefs, like the fringing reef here, a little bit on the patch reefs. And then when they went out to this barrier reef that we have here in Kaneohe, they saw little to no bleaching. In 2014, we saw that the bleaching was higher and paling on the barrier reef, and we saw variability and bleaching across the bay. We did see that there was high levels of mortality in the North Bay. And then 2015, we did this again, not as many sites as the previous year. And then 2019, we saw less levels of bleaching. So we just try to characterize how much bleaching are we seeing over time. We're seeing that the bleaching is decreasing over time, but we do see that the mortality is also increasing, except for 2019. So this gives us a snapshot of what's happening in the moment during bleaching. We try to go at the warmest time of the year, or prolonged, like the maximum amount of warming that they've experienced. That's always hard to tell as well. So the only way that we can really understand these dynamics, these bleaching dynamics in Kaneohe or anywhere else, is we need to have long-term monitoring data, right? This is essential, and we've been talking about this throughout this week. We do have some sites in Kaneohe and across the Hawaiian Islands that were established in 1999. So it's not extremely long, but it is a good amount of data so we can see what's happening in these areas. So the 3 sites that we have a northern site, which is the black line, the black squares here. We have a site in middle of the bay at Heia, which is blue here. And then the red site is located at the Hawaii Institute of Marine Biology, or Moku'oloi. So this is the southernmost site. And this is really helpful for us because coral reefs do go through these natural oscillations of changing their coral cover dependent on things that are happening. Sometimes the patch reefs grow and the corals fall off. Like there's a lot of factors that can influence this variability that we see. So it's important to understand if there's little to nothing going on that year, what do we expect to see the coral cover be and how that might change over time. So we wanted to really focus in on these last few years here from 2012 to 2019 to understand how coral cover is changing across these 3 sites. And I just want to share with you some interesting data that we've seen at these sites. So again, this is Ka'alaia, this is the North Bay of Kaneohe Bay. He'ia is right in the middle, like I mentioned before, and then Moku'oloi is at HIMB here in the southern part of the bay. And each of these different bars are a different year. So 2012, 2016, 2017, and 2019. And what you can see immediately is North Bay, Ka'alaia, it's relatively stable in coral cover over time. We see He'ia is declining over time, and Moku'oloi has declined quite a bit and is staying relatively low. And this was primarily concerning for us because HIMB or Moku'oloi is actually a no-take area, so it's an MPA zone. So we're interested in understanding, well, why are we seeing decline when there's little to no direct pressure on these reefs compared to the rest of the bay, which is open for fishing and recreation. So we started to look more about what was going on in terms of environmental factors in the bay. What's really interesting about this bay, because of the alterations that have occurred over time, it has influenced the residency time of this bay, and it actually makes it for a really interesting study system because you do have areas of high residency time. So here up in the north parts of the bay where it can be one day, the turnover of the water, versus the southern part of the bay, it can be up to 30 days or longer. So usually we see the water come in through the north parts of the bay and also through there's a channel here, or it could also come over the barrier reef and then out those channels. Once it gets down into this area, we do see that the water does hang out a lot there. We also put temperature loggers, which are these black triangles here at these different areas to try to characterize the temperature that we would see in the bay based on throughout these bleaching events. And I think that was the most interesting part, and I guess we kind of should have expected it. The area, South Bay here, with a higher residency time, had warmer water, sometimes up to 2 degrees Celsius warmer than the rest of the bay. And because of that, we did see that this reef has had higher levels of mortality and also declines in coral cover over time because of that warming that's happened there. So that gives us some insights into what are some of these local environmental drivers inside of the bay and how that might influence the coral cover and also coral resiliency, or how it recovers from these bleaching events. Also, so that's what we saw here at Moku'oloi, how this decline is likely because of the warming because of the long residency time that we see in South Bay. And it would probably continue to have higher levels of warming there through future warming events. And what was interesting, we saw at Ka'alaia, yes, of course, the turnover time and the temperature is relatively stable compared to the rest of the bay. But we wanted to look into more what is really going on here at Ka'alaia or North Bay with the coral cover. The interesting thing about Kaneohe Bay is it's really dominated by 2 species of coral. One of those species is Parietes compressa, or the finger coral. It's endemic to Hawaii, so found nowhere else. And then Montipora cavatata, which is a rice coral. And what's interesting is Parietes is usually the most dominant in the bay. So we wanted to look at if there's competition between these corals or a shift in the composition of the coral species over time. So in black here is the total coral cover in the black squares. The yellow triangles are Parietes compressa, and then the orange circles are Montipora capitata. And what we're seeing is that the coral cover is staying relatively the same, but we do see that the Parietes compressa is declining over time and that Montipora capitata is increasing. So we thought that was a very interesting thing where we see we just look at total coral cover, we might be seeing missing some shifts in the community composition, and there might be ways that one species is outcompeting the other or more resilient than the other, which gives us some insight of how we might see changes from this community shift over time, which is really exciting. So we're also looking into trying to understand, why are there differences between coral species and why are some coral species more resilient than others? So these are just typical biological measurements we take, try to understand, is it the symbiont? Is it the microorganisms associated with the coral? Is it something about the composition of the tissue and how the coral has evolved, or what kind of coral it is? And then also we have been extremely interested in understanding the boundary layer dynamics of the corals and the skeletal structure, how they build their skeletons. So we try to understand, if you have different shapes of corals, how are they modulating the water around them, and how is that influencing the environment that is directly the microenvironment that the coral experiences. So we've been doing a lot of microsensor profiling, and I don't have time to talk about it today, but if you have questions, I would love to talk with you all about this. So we're trying to understand pH and oxygen and temperature gradients as we get as close as we can to the coral, in the micrometers from the coral, and sometimes even touching it, and then moving away from it to see how the coral is modulating its boundary layer. And previous evidence has shown that if you go right to the surface of a coral, it is actually a degree centigrade or Celsius warmer than the bulk water. So these microenvironments could be explaining a lot of differences that we're seeing between species of corals, and also even within species of corals. So we'll sometimes see corals of the same species, one will bleach and one will not. And we're trying to understand, well, is that the biology or is that the microenvironment? So there's a lot of questions here to understand what's going on with corals. So of course, the understanding the dynamics of change in Kaneohe in Hawaii has been very interesting to me, and we're continuously doing work there. But I'm also located here in Texas, so this is where I am here in the Gulf. And we're really trying to take what we've learned in Hawaii and across the Pacific and understand the dynamics that are happening in the Gulf and the Caribbean. What's really interesting about the Caribbean is that it has had a lot of influence or declines over time. We have seen that the coral cover was relatively adequate in the late 70s, but we have had a lot of disasters or impacts regarding diseases, diadema die-offs or urchin die-offs, hurricanes, and also a lot of warming events that have left these coral reefs in a state of demise. Some of the coral cover is less than 10% in some of these areas. Some of them, we don't know what the coral cover is. We're trying to understand. All this red here is coral throughout the Gulf, and of course, just mostly the Gulf here, and then the Caribbean is further out. But one of the things that's really interesting is these shallow corals are relatively not doing so well, but we do have some pretty dynamic places that still may be viable for large species of sharks, also for sustaining fisheries, so we're investigating that. But we're also interested in understanding how some deeper water corals, and deeper, it's still, this is 60 feet to like 150, 200 feet. The Flower Garden Banks here, which is about 100 miles offshore of Texas, this coral reef is doing extremely well, and it's been relatively stable through these warming events and all these other impacts. And we're really trying to understand how these local drivers or differences in the environment might be influencing these corals, and how the biology of these corals are connected to other systems in the Gulf. So this could be a jewel that's kept moving forward, which we're excited to investigate more. So all this information, we're pulling this together to try to understand what are the impacts, what are the mechanisms of resilience, and how do we use this to provide solutions. So I mentioned earlier, we want to make sure that we're helping inform decisions. We're developing some research tools, and also, of course, training students and training the community so that they could help us with some of this work that we're doing. And I just want to briefly talk about informed decisions and the research tools. Earlier this week, throughout this week, we've been talking about marine carbon removal or ocean alkalinity enhancement. And Melissa and I are working on this project, trying to understand how ocean alkalinity enhancement might be influencing coral reefs. So this is ongoing work now that we're trying to do in investigating different minerals, olivine, calcium oxide, and sodium hydroxide to look at the impacts on corals. We're primarily focusing on Pacific corals, but we want to understand what are those impacts. Does it enhance calcification, or does it have adverse effects that we're unsure of? We've also been very involved in developing research tools that we can equip the community with so they can help us understand these ecosystems. And what I think is really powerful about this is that the community sees these declines, they want to help, but they don't know how to help. So what we've done is we've developed a color health card. This is modeled off of the Australian health card for Hawaiian corals. So this card is all the different colors of the corals, or the majority of colors of corals in Hawaii. And what's really unique about these colors is that they're all correlated with a physiological or biological parameter that is essential for coral health. So this gives us an idea of color means this in terms of the biology. And then we have our, we train our citizens, or the tourists, or anyone who's interested. We train them in going out and documenting corals to tell us if they're healthy or they're not healthy. So they'll do a simple survey of matching the morphology of the coral and also the color of the coral. So we want to know what relative color is the coral, so we know what the relative health of the coral. And then what we do from there is we have the folks go online, indicate where they conducted their reef survey. So we're zooming in here to Hanama Bay, and they provide all this information online regarding what the color of the coral was that they saw and what the shape of the coral that they saw. And from this information, we can gather, there's not that many species of corals in Hawaii, so we know based on the shape and based on the color what species it is. But they can also provide a photo of the coral as well, and other information regarding the depth, the temperature, so we know more information about that coral in that time point. And this allows us to have more eyes in the water, especially during warming events, so we can document where bleaching is occurring and where bleaching is not occurring, and then we can inform the local management agencies to go out and deploy a response team to look at these areas and to document bleaching if it is or is not happening. But this also gives us information at a broader scale of what's going on across the islands. And then lastly, of course, education and training. We're trying to equip students and also the public in these tools that we do use for measuring coral responses and also the environment around corals, so that they are better equipped for helping us in the future. And I just kind of want to wrap up here and just briefly illustrate the importance of why corals are important. Of course, they're essential for our livelihoods, but it's becoming more and more challenging for people to understand the importance of something that they can't see. And one thing that has been really useful is, and it sounds kind of weird as a biologist, but putting a dollar sign on something is a really great way to advocate for the importance of something. So this has been done with coral reefs as showing as they are an essential infrastructure or natural infrastructure for shoreline protection, especially during hurricane seasons. So that could be really helpful for advocating and for protection or restoration of corals, but also advocating for potentially how do we look at corals in the future, or protect them into the future. So overall, we estimate the total ecosystem services of corals is about $9.9 trillion per year. This can be broken into about $1.8 billion for shoreline protection, $36 billion for tourism, and then I think putting a price on culture and livelihoods is extremely hard. It's currently estimated at almost $6 billion per year, but over 500 million people depend on coral reefs. So just advocating for that is really important for people to understand. If we lose corals, then we lose because we're so tied to them and we depend on them so much. So just wrapping things up here and just getting back to this question of a rapidly changing system, I kind of wanted to think more about, can we slow the rapidly changing coral reef system? And there's ways that we could potentially do this. We can reduce the impact, and that's by, of course, regulation or changing human behavior to reduce that impact. We can investigate the resilience that we are seeing in some of the individuals and even across different species. So here we have the different pigmentation that I mentioned earlier within the same species. And then also we can develop solutions or provide solutions, provide training to people to help us document this decline, get them involved. And by getting them involved, you give them the responsibility to help take care of these ecosystems, which can be very, very powerful. And with this, there could be a potential for adaptation. We have investigated this. We have seen that corals can keep up with the amount of warming, but the rate in which the warming is changing, or the rate in which it's accelerating, corals cannot keep up with that. They have kept up so far, but they can't keep up to what we're expected to see for the future. So thank you again for letting me talk about corals, and I'm happy to take questions if I have any time.
Thank you so much, Keisha. That was fascinating. Yes, we have some time. So if anybody has some questions just for Keisha, we'll give her a few minutes, just like all the other speakers, and then we'll morph into the panel. There was one question in the chat from Laura asking, are you connecting with NemoNet to get citizen science information on coral cover?
No, I don't know about them, but I'm definitely going to check them out. Also, I'm going to play this video while we're answering questions. This is of the Flower Garden Banks if no one's been there.
Awesome. Okay, there's a question from Yuki. It's a long question, so I'm trying to read it. Okay, so before the military base was built in the region, do you think there was a greater water flow that might have helped to moderate water temperatures? And sort of how is human activity having an observable impact on the reefs?
So before, I would say that the alteration of the bay has significantly impacted the hydrodynamics of the bay. If those were not done, we would likely see that any types of land-based influence would have likely been flushed out of the system. But there's nothing we can really do about that now. In the 1920s and earlier, in early 1900s, the southern parts of Kaneohe Bay was described as a coral garden where people could take glass bottom boats to go see it. Now you would not want to go take a glass bottom boat to southern Kaneohe Bay because you can't see anything. There are some corals there, but not very much. So unfortunately, there has been so much change that it's probably irreversible regarding getting those back to what they used to be. I'm trying to see what else was on this question. Regarding current activity, the sandbar is pretty far from some of the corals. There is some coral near it, but likely not having a huge influence on them. But there are some people snorkeling in the area that do directly trample on the corals. Of course, that has an issue on, or that has an impact on them.
Perfect. Kristen, do you want to ask your question?
Sure. Yeah. Great talk, Keisha. That was very interesting. So I've heard from some other reports that having like more top predators on reefs, like sharks and things like that, like helps make them more resilient. So I was wondering if you could, so you could speak a bit about that.
Yeah, that's a really great question. And I would say also the opposite. When you see a shark on a reef, it is indicated of a redundancy in the system or a healthier system too. One of the things that with the top predators, even though I always like the joke because I'm like, it's always about sharks and whales, right? Let's talk about corals. But also corals are becoming charismatic too. But one of the things about the predators is that they're able to, they do help with disease and controlling disease of the different populations of the fish and other organisms on a reef, which helps maintain things in balance or helps keep control of one species from outcompeting the other. So there is a delicate balance that having the appropriate structure and top predators and mesopredators and so on is essential for maintaining that health of the reef as well. What we're seeing at the same time is that if you lose certain species of corals, there are certain types of fish that are only in other organisms that are only associated with that coral species. So then you have that cascading effects of losing that coral with that specific fish or other animals associated with it and so on. So that's also very scary of what we're losing individual corals that we don't even know how that's impacting the rest of the food web.
Thank you. And in the Hawaii reefs you spoke about, are there a lot of sharks and things like that?
There, in the main Hawaiian Islands, I have not seen that many sharks, unfortunately, with all my time spent there. In the northwestern Hawaiian Islands, where there is less human influence, there is more sharks, quite a bit of sharks there. I think there have been some documentation to try to understand the differences in the shark populations between the northwestern and the main Hawaiian Islands. It's some people attribute it to human presence or, or just lack of impacts in those areas or fishing, of course. So it just depends on what are the factors that are driving that. But I had to show the video of the hammerhead because that was the first time I've seen a hammerhead in Mexico. It's like, that is so cool.
That is awesome. Thank you.
Thank you so much, Keisha. Now let's morph into our panel. So if all our speakers from this morning can turn on their videos, and everybody else on the call, feel free to ask questions. You can raise your hand and ask questions or put them in the chat. These could be for specific speakers, but they can also be sort of more general themes that you would like any of the speakers or multiple speakers to answer. We also have some questions prepared, so if the audience members don't have questions ready, Kristen, Melissa, and I are ready to go with some questions. Okay, well, while people are thinking, this is a question sort of we have been thinking about in advance and then came out of some of the chat and questions. I think it was to Tricia, but it's really to all of you. You've all talked about a lot of variability, whether it's the Northern Bay versus the Southern Bay or these microhabitats or the different parts of the WAP. How can we sort of both quantify and observe this within the context of rapid change? How do you observe rapid change if everywhere you look it's affecting differently? And then also how do you present this in terms of sort of informing decision makers? Whichever of you wants to start off.
I'll just start and just say that we need to embrace the variability. So I'd like that was brought up in the previous talks, is because we tend to, like, as biologists, we mean everything, we average everything, right? But we're seeing that the response is actually because of the extremes in the variability. And that's something that we need to capture. And I think just because it becomes more convoluted and harder to explain to people, I don't think that we should shy away from talking about it and being able to explain that the variability is important, because in the biological aspect, it shows us that there could be selection that's happening, right? But in the environmental aspect, it shows us that the variation that we're expected to see is going to get worse, and that's going to have even more impacts. So that's kind of what I'm just like, I'm always like, we need to talk about variability, like, constantly.
Yeah, I think that's a great point, Keisha. And just, I think in some ways too, it's almost like recognizing that these systems are constantly changing, and maybe it's a way that we're communicating this change. We have to reframe it in that, like, there's always going to be this variation in any system, and kind of better showing the extremes of that variability when we're talking about a system that's shifting. And then, saying this is kind of the average change we see, but really this can vary from X to Y. And I think encouraging that there's like this more nuanced variability is really important. And I think even that comes down to like when we educate people about talking about this variability. Like, I just think in my classes too, like, students so often want to have like, what is the answer? What is the answer? And it's like, well, actually it's this range of options. And so maybe posing it a bit more like that could be helpful. I'd be interested in getting Emelia's perspective too, just with her ice communities and whatnot.
Yeah, well, no, I think I agree with both of your points. You want the end members, right? You can't really constrain a system until you know its wildest variation. So that's kind of fun in a way, thinking about that, of like, how far can we push the system? That might also then give you an idea about change, right? So you need to have those end members in order to have a meaningful mean.
Thank you, Debbie. I saw your hand up a moment ago. Do you have a question?
Yeah, thank you. Thanks for all those great talks. I just had a comment about variability, and I absolutely agree how important it is to communicate that. But I also think we have to remember who our audience is, because when Tricia's up there testifying to Congress, they're not going to understand variability, right? We need to have clear messages about, yeah, there's variability, but the long-term trend here is X. And, we have some real concerns about the health of this ecosystem. So, yeah, I just wanted to comment about, it's important as scientists, and we understand that, but most people don't. And especially at this time, we have to be very careful about sending clear messages. Just wanted to mention that.
Thanks. Masha, do you have a question?
I do. Great session. Thank you. Thank you all. I have a question to Emelia, and you might have talked about that, but it's early in the morning, in California, so I might have missed it. So about 10 years ago, there were several studies, and specifically I'm thinking about a paper by Kevin Arrigo, who argued that because the sea ice is melting, the productivity overall is increasing in the Arctic. And you talked about the smelt ponds, right? So overall, obviously, there should be overall higher productivity in the melt ponds and under ice. So can you talk a little bit more about that in this, in this sense?
Yeah, no, that's something I think about a lot because I think, that paper, I know exactly what paper you're talking about too. That's a very like open water versus sea ice perspective. So it's like, yes, you have open water, so there's more chlorophyll that you can see from satellites, but we can't see what's under the ice from satellites. So when you're just doing a chlorophyll equals productivity measurement or a satellite measurement like that, you're not capturing what's happening within the ice. And we know now that we've done so many of these drift studies and taken under ice light measurements, that there's a great paper out recently from Clara Hopp at the UABC that shows that sea ice algae can actually grow at much lower light levels, like below what we consider the minimum light requirements for growth. So there's actually a lot of productivity underneath the ice, in the ice, that I think is not necessarily, we don't have it captured. So it's really hard to say, oh, open water equals more productivity, without knowing what the ice productivity is to start. So yeah, maybe overall values, but for sure the community composition will also be different. So we don't know the impacts of that either.
Yep. Thank you. Great point. Thank you very much.
Melissa?
Yeah, thank you so much for the great presentations. Maybe this is for Tricia, but maybe for everyone, this is related to the variability. And I was just wondering, how do you deal with these extreme years, like maybe the phytoplankton bloom that you saw in the Gulf of Maine in 2023, in your long-term kind of like time series analysis or trends?
Yes, they can be very annoying when you're trying to statistically analyze them. Which, I spent a long time trying to figure out how to really analyze, especially this estuarine system, these long-term changes, because they were so variable. And I think like the figure I showed for the Narragansett Bay was kind of a nice summary in that we used some pretty complex modeling that allowed, based on like Bayesian statistics, where like it allowed us to kind of use the previous year's data to help inform the current values that we were modeling, and that helped us to kind of like still incorporate some of that large variability, but it did help us to kind of like constrain it a bit more than before. But then we also looked at some of the raw values when we were looking at like the cumulative chlorophyll A. That wasn't modeled data, that was just like observed data, which kind of helped us to include more of that natural variation that we saw, because that was something I struggled with a lot, was just like wanting to showcase that variability, but then also wanting to be able to model it and analyze it in a robust way, which oftentimes outlier extreme data like that's very difficult to do, to work with. So that was kind of how I got around it. But and sometimes, I think you can also showcase where you include a model like without some of those outlying extreme data points, but then you include it with to kind of show how the model changes its output. But anyway, I think trying to kind of incorporate a variation of different ways that you look at the data can be a helpful way to kind of show how you account for all those varying data points.
Do Amelia or Keisha, do you want to add anything to that?
I'll just say, usually when we have extreme like temperature events, it's when we have a bleaching event. So that works out for us for characterizing it. Which I was just looking at our sea surface temperature graphs in Hawaii, and it's every time you see some variation in warming, there's a bleaching event that happens. So for us, it's just taking that variability, that response, and comparing it over time. So that's how we capture and see. The hardest thing we have is like the degree of variability, like how much did we see and what was that impact? That's something that we're trying to get our mind wrapped around, and we usually do that with degree heating weeks. But then I also think that we're not capturing some accumulation of heating over time too. So it's like a shifting baseline, really, for the responses that we're measuring.
Suzanne, do you have a question?
Yeah, Debbie inspired me to maybe suggest that variability, we're using that word in multiple meanings here. So your uncertainty in your temperature measurements are minimal. They're a fraction of a degree. And so there's no doubt that the ocean is getting warmer. But on top of that, you have extreme and unusual events that may have unique impacts. And some, phenomena like this dinoflagellate bloom are difficult to explain. And my question earlier was more directed at how do we get at expressing these extreme events like, a shelf-wide dinoflagellate bloom in averages. But that's not to distract from the fact that, climate change is happening much more rapidly, and the data and statistics fully support that. So to be mindful how we use uncertainty and variability.
Yeah, I think Keisha's point sort of makes me think about thresholds and how, like she's thinking about bleaching, and a lot of what I thought about too with the Narragansett Bay data was like bloom magnitude, like when we reach these like really large blooms, kind of like what Suzanne's talking about with the dinoflagellate bloom, it's like, where do you define that threshold? And I feel like that's almost like a separate analysis to looking at like the long-term trend in some ways. But that's just something that Keisha brought up that made me think of that I'd point out.
And just to talk with that, Tricia, like we established thresholds, and we also are trying to see, are those thresholds changing over time too, which adds a whole nother layer to things. But that's how we're trying to quantify like corals keeping up with the warming. Those are all good points.
I might just jump in too to think about. I like the idea of the changing baseline and these thresholds. We don't know about their changing, but I think a lot of that also comes down to, particularly for bloom dynamics too, we don't know the mechanisms necessarily that are triggering in a lot of ways. So I feel like there's still a lot of groundwork to be done. We can correlate it with the variability that we're seeing in the environment, but if we don't know what the mechanism is, we don't know what the trigger was. So it could be the heating, it could be something else, it could be cofactors. So more of those types of like mechanistic work too, I feel like is important to constrain that, to make it go from uncertainty to this is natural variation.
Branching off from that, do you guys think we need some new observational tools or modeling tools in order to observe these rapid changes more quickly and understand them maybe more definitively? And if we do, what tools do we need? And if not, what tools do we have that you think are working really well and maybe we should exploit further?
I'm all ears. Love to hear your thoughts on tools.
For us, for coral stuff, it's about resolution. Like how high quality data do we have? Like right now with sea surface temperature data, we have 1 point for all of Kaneohe Bay. And that's really challenging to understand how that might be influencing response if it's just such a large area. When, I just showed like we have like reef specific responses that are happening and warming. So that's like ours is getting that resolution to understand temperature and other factors in these areas that, a finer scale.
Something that we realized with the big tripos bloom that occurred in the Gulf of Maine was that we didn't really have a good mechanism for communication about when that happened. It ended up just being this like giant email chain among people throughout New England of being like, have you seen this bloom? Have you seen this bloom? Have you seen this bloom? And, because the satellite record only shows the chlorophyll, doesn't show the community composition, although maybe with PACE, we can help figure that out better. But I think that was something we realized, at least in New England, was like, we need to have better mechanisms for communication about some of these potentially extreme events, because otherwise we're all just kind of in our tunnel doing our thing. And so I think that's something that we can definitely continue to work on as a community.
I wonder if OCB could help with that. There could be some like extreme events networking site or part where people could put stuff in and other people could check.
That's a great suggestion and something that we actually talked about when we had the extreme events workshop back in 2022, I believe it was. We talked about the need for kind of this grassroots coordination effort, leading up to, through, and then after an extreme event to be able to capture some of that baseline, capture the pulse of whatever it is you're measuring, and and then the recovery. And that's, we talked about, we had a little, we put together a document for the Ocean Climate Action, OCAP, Ocean Climate Action Plan. And we provided some recommendations, which we can share that in the chat. But one of the ideas was kind of these little, leveraging existing infrastructure in places that are rapidly changing and experiencing these extreme events more frequently, and having a network of them, almost like an LTER, but an extreme events network, where we could leverage existing infrastructure and get people organized and coordinated.
That's really cool. Other questions? I have one question. When you go into, I just feel like I'm asking all the questions, so please, other people can answer that. Kristen and Melissa helped craft these questions, so it's not just me. Oh, look, there's another question by Jerry. We'll do that. Is there any information on the corals of Bering Sea in Alaska and the effects of trawling by the 350-foot trawlers scraping the bottom, destroying the habitat? Do you know anything about corals in Gulf of Alaska and Bering Sea?
Yeah, Jerry, I meant to look into this before talking today, because I did want to talk more about deep sea corals and whatnot, even though it's not my area of expertise. But I think it's not good. Like trawling has been shown to be extremely problematic, and I think we talked about this before. And with potential mining that might be occurring, that's one of the concerns we have in the Gulf as well, the Gulf of Mexico. Because there are really well-developed deep sea corals here, and with extraction practices, it likely will have a huge impact on that. So that's definitely an area that we need to understand more. And it's just one more of those. The hardest thing is documenting something that it's hard to document or see. And going deeper is usually a lot more challenging to know the effects of things if we don't have that previous data, that long-term data. And also just getting there and documenting it is extremely challenging.
Thank you. I very concerned with the trawlers in the Bering Sea. I was out there in 1992, and one of the trawlers of 60 boats caught 60,000 pounds of coho in 60 minutes. 15-minute drag, and that's scary. But even more concerning is what you were just talking about, coral reefs and the habitat. That's our nursery out there. That, everything that we depend on comes from the ocean, pretty much. And the coral reefs provide habitat for the small fish so the big fish can eat them. I'm singing to the choir here, the importance of coral reefs, and the trawlers have been trawling since 1982. Since the 1960s with the international governments, but 1982 with the North Pacific Fisheries Management Council. So that's 43 years of trawling on an annual basis, and they do it year round. And these guys that are bottom trawling, they get 50 ton of product in 30 minutes. And that product sinks to the bottom. So when they're dragging that around, it's just a big old bulldozer that's, 15, 16 feet wide, that's crushing everything on the bottom. And you do that for 40 years, and you're going to end up with nothing. So the habitat of our fish that we depend on is being destroyed by the corals. I really like your presentation, because that brought to me my attention the importance of habitat and the importance of protecting the place where they go grow up, and come back to us. And pretty much everything is dependent on them. The corals, the fish that we eat, the marine mammals, everything. And I'll speak to that later. But thank you very much. That just brought home vital importance of habitat and the coral reefs. So I just know that in Alaska, it's been, 43 years now that they've been scraping the bottom. So 43 years of bulldozing, and what do they know of the effects of coral reef destruction? Or, thank you very much.
Thank you, Jerry. And just to add to that, the deep water corals don't have the algal symbionts, so they grow a lot slower. So removing them likely means they're not coming back because it just takes so long for them to grow. So I appreciate you mentioning that and talking about that, and also that knowledge that you have. Like if it's not documented in, like with long-term monitoring or stuff, it's really hard to get that information. So it's so important to have this local knowledge so that we know more about these systems and what they used to look like, because otherwise, if we go look at the deep sea corals there, we would be like, oh, there have never been any here, and not even understand the effects of trawling if we didn't know that. So thank you for providing that information.
Exactly. I mean, I wish that we had a baseline of the corals way back in 1982, so that we can go back now in 2025 and just to show how much damage there has been done to the corals in the Bering Sea. But that's something that we don't have, information that we don't have. That's academia hasn't brought that as a concern. Maybe because the red flag hasn't come up. The red flag is up. We need some help in the Bering Sea. And whoever or wherever this help would come from, I would welcome it. And thank you for your information on the coral reefs.
Of course. Thank you, Jerry.
And thank you so much to all of our speakers from this morning. Our time for a panel is about over, but thank you to Tricia and Emelia and Keisha, and also Lourdes and Maya. It's really been a great morning, and I've learned so much.
Quite an exciting update from Dwight. All right. So we have an exciting lineup of 3 plenary talks today. This afternoon, we're probably going to follow the same format at this morning. So we will have a panel discussion with all of our speakers at the end, but if we have time, we'll take 1 or 2 questions after each presentation. Otherwise, all of the questions can be saved by the end for the panel. So this section brings together diverse perspective on ecosystem change, from the modern impacts of marine heat waves to traditional knowledge from the Bering Sea, to a paleoclimate event that represents the most dramatic example of carbon release and warming in Earth history. These talks will help us better understand how today's rapid changes compare to those that have occurred over geological time. So our first speaker is Zach Gold, Group Lead of the Ocean Molecular Ecology Group at NOAA Pacific Marine Environmental Laboratory, or PMEL. Today, he will present how marine heat waves impacts fish population in the Pacific Northwest using time series data on zooplankton and environmental eDNA. So take it away.
All right, thank you so much, and thanks everyone to be here. I appreciate having Dwight here. We work really closely with the Ocean Acidification Program, as well as all of our other OAR colleagues. First, just want to start off that there's a ton of co-authors on this work, and that this work is really an evolution of the last 8 years of this sort of project that sort of spawned as a Hail Mary as a graduate student, and now that had the opportunity to pursue this research as a federal PI at NOAA has really been a dream. I'm coming from the NOAA Omics program, and so for folks that aren't familiar, please go to the NOAA Omics website. But NOAA is trying to leverage, the rapid advancements in biotechnology to, basically be able to do our agency missions better. And so, today I'll be talking about one element about eDNA, but across the agency, NOAA is using new omics techniques to be able to, answer questions better, cheaper, and faster. And really today I'm giving this presentation standing on the shoulder of giants. And so the Cal Coffee program, which has been going for 75 years, much longer than I have been alive, even than my parents have been alive, to answer some really fundamental questions about how our marine ecosystems are changing over space and time, and to make that information relevant for agency and management decisions. And so, the California Current, in thinking about this talk, I really come from the opposite of this sort of the OCB. I usually think about how biogeochemistry is changing, and then that impacts the marine ecosystems. And I don't often think about, it's not my background to think about how the marine ecosystems themselves are actually changing the biogeochemical cycling. And so, often we're thinking about how do multiple stressors impact on marine ecosystems, but obviously we know in marine ecosystems, the biology is a key component, are changing the underlying biogeochemistry. And so what's kind of made me think really hard about this talk and how to frame the work that I have been doing is thinking about, this bottom up and this top down of where marine species, play really key roles in our biogeochemistry. And I'm going to talk about both of these today. It'll be sort of more on the bottom up impacts of changing biogeochemistry on marine species. But hopefully it can lead to new directions in the reverse of this. And thinking about how some of the new data sets are bringing online could help us answer some of these bigger picture biogeochemical questions. And I just want to start with saying the California current has changed for a long time. This is data from 1992, also before I was born, looking at archival Pacific sardine and northern anchovy scales from sediment traps in the Santa Barbara Basin, and producing almost 2,000 year time series. And there have been really big booms and busts of anchovy and sardine. And obviously, if you go back to basically time immemorial, we know that the California current has changed, a lot. And so I think one thing that is, common in this really dynamic temporal upwelling system is that change is constant. But this, background noise is obviously on top of the fact that we are accelerating our climate change and changing our biogeochemistry in ways, that 100 years ago we could never really dream of. And so in a more temporal and recent example that led to the formation of the Cal Coffee program, is, the sardine fishery collapse made famous by Cannery Row in 1948. And, 1940s that led to, in 1948 and 49, the formation of Cal Coffee with their first survey in the 50s. And, the big question there was what drove, the demise of and the loss of the sardine fishery that at the time was, a huge local economic engine, but completely disappeared right after World War II. And so, Cal Coffee started this gridded survey. This is the full extent survey. They typically only go to, just above point conception for half the year. They now do quarterly cruises. They used to do the entire U.S. West Coast monthly, and they had 12 dedicated ships to it, which is unbelievable. But in 1984, that funding was cut. There actually was a year without Cal Coffee cruises. And so, Cal Coffee itself as a program has undergone rapid change over time. And it's led by NOAA Southwest Fisheries Science Center, CDFW, and Scripps. And one of the fundamental questions that was underlying the formation of this program actually came from a 1914 manuscript. And this is, a 2008 paper reflecting almost 100 years later on it. I was thinking about this critical, period where, for fish and a lot of fishes that do broadcast spawning, they will produce on the order of 100 million to 8 billion eggs. And only 2 on average end up surviving to keep the population going. And so there's this huge critical period where the vast majority of, larvae and eggs are disappearing. And at the time, in 1914, this was a novel idea that the environment and the biogeochemistry was driving, who are the winners and losers. And so Cal Coffee program was built on this, and it was one of the first large-scale programs, and it is the longest running marine biological oceanographic time series in the United States. They're slightly bitter that there's one in the UK that started 6 months earlier, and we unfortunately can't change that. And this has reflected this kind of work over and over again. This is obviously a more complicated, vision of the previous slide. But to sort of break it down is, there are changes in the biogeochemistry, there's changes in the metabolism, there's changes in the prey availability that are going to lead to sort of the success of baby fish as they go through this life. And so, one of the fundamental questions that Cal Coffee's been trying to answer for a very long time is, how can we predict this, right? Because if we can predict this process, then we can do a pretty good job of predicting our fisheries recruitment. We can know exactly how much fish we can take out in a given year and not drain the bank. And obviously this is, the holy grail of biological oceanography and fisheries management is to be able to answer these questions. And we really haven't had the capabilities or tools to do this until recently. And so, 75 years on, we are still answering the same questions, but our tools and capabilities are now here with the technology to be able to answer, these questions that we just didn't have the capabilities to do. And so now I'm going to do a sort of blitz of how the, California current, and this says biogeography, but it should be biogeochemistry, is rapidly changing. And so, we're seeing warming. This is the last 40 years of warming. We're seeing pretty rapid warming across the California current. Certainly since 2014, we've had, I believe, about 7 of the, 10 top hottest years on record. We can see in the marine heat waves are becoming more frequent and larger. And so, if you look at just the last decade here, there's quite a few of these. Certainly the blob was off the charts, but there was another one in 2019, another one in 2022, and another one in 2023. And so we're in a little bit of a lull here, but, obviously they're becoming, larger and more frequent. We're also seeing intensified upwelling conditions. We're getting stronger winds, and so we're getting, greater upwelling. Often this leads to greater differences basically between, who's in the upwelling zone and outside. We're also getting, increase in eddy formation, which is really interesting, and how that, might change, the biogeochemistry, regionally across California current. And obviously there's differences, from between Washington and the central, versus the southern, large biogeographic breaks and differences in the physics happening there. And obviously we're seeing changes in ocean acidification as well. And so, we're seeing, work from PMBL and the West Coast Ocean Acidification Cruises, looking at multiple decades now of declining pH and aragonite saturation state, across the U.S. West Coast. And we're also getting deoxygenation, just to keep piling on here. I promise it will get less sad in a second. And so the, combination of all of this, on top of the fact that in parts of the California current, particularly in Southern California, most people might not know this, but Wilshire Boulevard is the population center of gravity in California. There's 25 million people that live in California. And actually, it turns out 25 million people and their sewage waste really can alter the biogeochemistry. And some really incredible work that's been coming out of UCLA and Princeton, University of Washington, and SCORP, now sort of leading the charge here, has been showing how this change in nutrients is changing how much phytoplankton are available, is changing subsurface respiration, and is leading to these, pretty remarkable declines in habitat thickness, particularly captured in these island eddies off the Channel Islands, where we're seeing, an upwards of seasonal loss to 25 to 30%, 40% of sort of the habitat thickness and available habitat for marine species. And we don't actually know what those impacts are yet. This is, the last sort of 5 years worth of work to be able to pull together these incredibly accurate biogeochemical models to be able to look at things over space and time that we really haven't had the capability to do with observations. And as these models get more accurate and more validated, we can see sort of the extent, of these problems. And so this is the landscape of our rapidly changing biogeochemistry across the West Coast. And that's going to impact, sort of which fish and how fish are reproducing, and the winners and losers of not just fishes, but obviously the zooplankton and the phytoplankton and the microbes. And so just to summarize, here, obviously, the biogeochemistry of the California current has changed quite dramatically over the last 100 years. But at the same time, as our biogeochemistry is changing, our tools to be able to answer some of these questions of why and how biogeochemistry matter are increasing. And so, just highlighting in this space some really, sort of incredible work on looking at metabolic index theory and basically linking hypoxia tolerance and temperature tolerance together mechanistically. And obviously, the ability for water to hold oxygen is linked to temperature. And looking at these differences in slope, we can start being able to basically predict where species, should be based on their temperature and oxygen sensitivity. And there's been some really incredible work, including some Evan Howard here at PMEL before he got here, looking at how, looking at the basically habitat volume that is predicted, by metabolic index theory, does a pretty good job at predicting the larval abundances of anchovies over time. And so, being able to take this same kind of model, apply it to multiple species, and answer how, basically linking how the changes in biogeochemistry are leading to, you know, the changes in fishes from a mechanistic, you know, modeling approach and theory is really critical. And we didn't have these, you know, ability to do this. And obviously this leverages, you know, lots of incredible painstaking work in the laboratory to be able to calculate this respirometry data to enable this to happen. And, you know, other really incredible advancements on the technological side are ability to use stable isotopes to be able to go back in time and look at how energy transfer efficiency and food chain length have changed over time. And so, you know, this was a really great paper by Rasmus Swelthop at the Cal Coffee program and many others, basically being able to, you know, look at times of boom and bust of anchovy and looking at the food chain length. And one of the things that's really interesting is it appears to be, you know, linked to biomarker signatures of heterotrophic protists with this idea that, you know, these shorter food chains lead to, you know, more food availability for the larvae, which leads to more adult anchovies. And this sort of boring out and the importance of being able to link all these things together. Of course, that gets down to the question of, well, who do baby anchovy eat? And as you can imagine, a larval anchovy is unbelievably tiny. They have straight guts, so it's pretty hard to work with them. But, you know, our ability to look at the anchovy water, who are the larval anchovy interacting with, have really exploded with our, you know, new tools. And whether that's from the NOAA Cal Coffee Ocean Genomics Program, NCOG, you know, looking at changes in microbial and phytoplankton assemblages, or, you know, being able to combine that eDNA, you know, with the presence and location and abundance of anchovy larvae, we can start to disentangle this. So this was some really interesting exploratory work, you know, linking these together to be able to identify, you know, both prey and the microbiomes and the potential predators of anchovy, you know, with thousands of species, you know, strongly correlated. Obviously, it's difficult now to tease those apart, but it helps us narrow down, you know, who those potential prey targets might be, and put a, you know, a name to the face of some of the phytoplankton players that seem to be, you know, correlated with strong anchovy abundances. And so where I sort of have come in this sort of experiment and, you know, really a Hail Mary sort of trial of, hey, what happens if we took a jar of zooplankton that are collected on Cal Coffee since 1996, and we, you know, pipette the ethanol and we pretend it's an eDNA sample? Can we reconstruct who's living in the jar and then, you know, go back and reconstruct, you know, the entire, as much of the Cal Coffee time series has been preserved in ethanol as possible. And so I will not bore everyone here, but please follow up with me afterwards about working towards how do we get abundance data, you know, using metabarcoding and amplicon sequencing reads combined with other sources, and, you know, really building off of Oli Shelton and Ryan Kelly's work in the space where I did my postdoc. And, you know, what we're able to show in this work is that we can do a pretty good job from, you know, joint modeling the counts of some of the species of ichthyoplankton that are painstakingly identified by the expert taxonomists at Cal Coffee with the metabarcoding data, which is identifying more species, including a bunch of species that can't be morphologically identified because they're too small to get, you know, down to species level resolution. And, you know, together, this allows us to track a broader suite of, you know, the marine or the species that are being captured in the Cal Coffee tows. And so one of the big things we found here, and this was data, you know, that we found, but also borne out by the morphological counts and a bunch of other, you know, sort of independent observations from a bunch of different cruises, is that the marine heat wave drove these really novel community assemblages. We weren't seeing just, you know, mesopelagic southern species entering in. We were seeing these mixed communities of both, you know, the species that are, you know, sort of cold associated and warm associated, all at them at the same time. And these novel species assemblages had never been recorded in the previous 70-year time series. You know, usually we would get, you know, just the warm species or just the cold species, and we wouldn't get these really, you know, diverse, actually the highest diversity of ichthyoplankton that had ever been observed. And so this kind of really threw a wrench, because if you went and you looked at all of the time series data prior to 2013, you would say, all right, anchovy do really well in cold water and sardine really do in warm. Marine heat waves off the charts. We expect sardine to do great, and they didn't, right? All of our previous correlations failed. And so part of that appears to be that we are just looking at a simple correlation. And we didn't really have a true mechanistic underpinning of the multiple stressors and the biogeochemistry that were happening. And it turns out that sardine, yes, have higher temperature tolerance, but they also have a lower hypoxia tolerance. And so that suggests that actually the interplay here, and just from the metabolic index theory, we would, and the rates for these species, we'd actually expect that if there was lower oxygen, anchovy would win in that situation. And so this suggests that we need more of these mechanistic and holistic ecophysiology models to be able to predict, the winners and losers. And this is not surprising to many folks, but I think, being able to integrate this into how we think about how species are going to react to climate change is going to be really useful to give us some of the tools to maybe be able to predict these black swans, right? And sardine, disappearing when temperatures got warm was certainly a black swan for the fisheries community at the time. And so now it brings us to today, which is, this project that we've been dreaming up and scheming, and I wish I could be presenting to you on the data yet, but we're actually just hitting the ground running starting next week. And so sort of teasing the work that we're leading up, which is we were able to secure some funding to be able to basically pull together a 25-year time series, 25 stations and 2 seasons to be able to answer these really big, questions of how zooplankton are changing over space and time, and hopefully getting towards, this idea of a climate grade zooplankton time series for the Southern California current. And there's been incredible work studying zooplankton in this space, and obviously, lots of great work with Moira Decima and Lindsay Sala now, and Mark Oman before them, and Andy Thompson and Edward Britton before them too. But there's just not been enough time in the universe to count anything more than krill. And so being able to actually identify thousands of species simultaneously is really where eDNA comes in the clutch here. And so here's just some pilot data that we got back, where we're able to show, that, one, we're able to get a ton of species, and 2, we're seeing, not surprisingly, different communities and different temperatures. We're also able to use the same kind of models here, just looking at the krill data, where we are able to do a pretty decent job. I'll say we didn't have a lot of data points in the pilot data, to start being able to start fitting these models to estimate abundance estimates, which is, really helpful, especially because we are only going to have, krill counts from spring when those data exist. And so being able to sort of, use these joint models to anchor the predicted abundances is going to be really important when we don't have any counts from fall. And so we see similarly, just looking at the krill data, that we also saw, changes in the marine heat wave, and here with mesopelagic southern species, dominating and showing up far more abundantly. And, the Nyctophany simplex typically only shows up in the California current during El Nino years, and they've seemed to have stuck around at least for the last 5-6 years, which is really interesting and suggesting, that the system is changing as it is getting warmer, as we would predict. But, obviously there are some places where we are seeing similar novel communities of multiple species of krill, typically that don't, overlap, but both being, abundant, which is, really interesting and has a lot of potentially, interesting downstream effects on, how the ecosystems are going to function. And the other thing, in addition to krill, is that we were getting, obviously, we got almost 1,000 species identified to species level, which is pretty incredible. This includes, market squid, spot prawn, and rock crabs that are harvested. Market squid in particular is one of the largest state fisheries along the West Coast, state-managed fisheries. And, being able to basically provide an independent estimate of some of these spawning stock biomasses might be really critical for helping states manage their fisheries. We also get, pteropods. And so we detected and identified 9 species of pteropods and listed a couple classic ones here. And, another story that I'll just sort of highlight that's kind of a tangent, but I think relevant to how our, California current ecosystems are changing, is we detected sea stars. And their larvae are pretty gooey, and they usually don't survive the net tow very well. They usually dissolve on contact with ethanol. And same with jellies that we detected a ton of. And so this suggests that we can expand, who we're capturing. We actually don't even need to be able to see them as long as their DNA was caught in the net. And so this lets us look at, a lot of the other underappreciated species, like the hydrozoans, like the larvaceans, like the jellies, and like the benthic invertebrate, star marrow plankton that are often, severely undercounted and not looked at systematically. And so we look at just the STAR data. One of the things that's, really interesting here is that we detect Pictopody helianthoides and Pisaster giganteus, the sunflower star and the giant star, up until the basically onset of the marine heat wave, and we never detected Pictopodia larvae again. And for people that are not aware, Pictopody helianthoides, lost over 94% of the population because of sea star wasting disease that occurred with the onset of the Pacific marine heat wave. And so this suggests that, the Calcopy ichthyoplankton or zooplankton data is also being able to provide, information on benthic invertebrates and their standing stock biomass. And so obviously this has big ramifications for the state of California. About 300 kilometers of coastline, basically turned into urchin barrens almost overnight with the loss of one of the major urchin predators. And obviously this had, pretty severe changes in itself to the biogeochemistry with the loss of kelp. And so, losing these keystone predators, can obviously lead to large-scale changes in marine ecosystems. And so one thing that we've been working on sort of related to this is basically developing a sunflower star eDNA assay that we just demonstrated works, in the field, from some transplantation work at Friday Harbor. And so now we have a tool to be able to go out and try to find the last of these individuals and hopefully can help, recovery and management efforts for this iconic kelp forest species. So that brings me sort of back to, we hopefully can use this, climate grade zooplankton time series to not just answer questions about the zooplankton themselves, but also benthic invertebrates. But also, I think, increasingly thinking about how we could use this for answering, a series of other questions, right? These are prey fields for fisheries and protected species. Krill are the favorite food of many marine mammal and baleen whale species, but this could also be really useful for looking at leatherbacks and seabirds that are, we don't have good information on their prey fields. And obviously answering those questions of how is coastal eutrophication impacting, these coastal pelagic habitats, and how is changing ocean acidification and marine heat waves potentially influencing, the winners and losers of our zooplankton communities. But we can also do the inverse. And this is kind of where I think this might be an interesting conversation. Happy to follow up about this because it's not really my space or how I think about, these kinds of questions generally, which is, could we leverage this 25-year, zooplankton time series to answer questions about biogeochemical processes? And obviously zooplankton play a really critical role in the carbon pump. And oftentimes, though, we call them large and small zooplankton in our biogeochemical models, and that's the end. But obviously we know, there's 15 phyla of zooplankton, at least, if not more, with very different taxonomy and ecology, and those are going to have big influences on particle size and transport. And so could we, link these data together to answer these bigger questions? And, also from this incredible modeling work that's being done, we can also answer different kinds of biogeochemical questions, right? There's questions right now we're thinking about intentionally changing our local biogeochemistry with either water recycling or marine carbon dioxide removal. And so can we, use these, digital twins model world to be able to answer how, might we be changing our biogeochemistry and how that might impact the zooplankton species and how the zooplankton species in reverse might impact the success or of these intentional biogeochemical changes. And so sort of end here a little bit, I think I brought this point up before, which basically, can we leverage a 25-year time series of zooplankton to, do a better job of moving beyond just large and small zooplankton to, put more names to faces and more finely resolve, how the individual species and their biology impacts our biogeochemical and large-scale carbon cycling. And so with that, I'll leave it here and probably went a little over time, but thanks for listening.
Thank you so much, Zach. We'll maybe pass questions to the end, and let's move forward with our next speaker is Jerry Einabov. I'm not sure if I'm pronouncing his last name very well, but he's an indigenous elder and lifelong hunter and fisherman from Alaska. He will share his perspective on the ongoing ecological crash in the Bering Sea, offering insights rooted in the indigenous observation and stewardship. And I'm just gonna let Kristen probably turn on and talk a little bit about how Jerry got involved in this section.
Sure. Thanks, Melissa. So yeah, it's a real pleasure to have Jerry Ivanoff here. Yeah, as Melissa said, he's an Indigenous elder from Unalakleet, Alaska. And for those who don't know, the term elder really denotes like a lot of respect from within his community, as well as a lot of wisdom. So I think that's just an important point to make. And I met Jerry through a project at NCAR that we have called Rising Voices, Changing Coasts. And this aims to merge Earth system sciences with Indigenous knowledge. And well, it's been a challenging project. One of the things I've learned and have been really impressed by is how intimately tied the Indigenous communities are, especially in Alaska, with their ecosystems. And so they're actually like part of the ecosystem, rather than in Western culture, we tend to think of humans as more separate from the ecosystem. And so Melissa and Rachel and I really thought this would be a valuable perspective to add to our rapidly changing systems session, and in particular, just to bring Indigenous knowledge to OCB. So yeah, with that, Jerry will be talking about the ecosystem crash in the Bering Sea, and take it away, Jerry, if you'd like to start talking, or put your camera on and start your talk. That'd be great. And yeah, Melissa's got the slides that are the pictures that you sent for the slides.
Thank you, Kristen, and thanks everybody for your presentations. Really interesting. I'm Jerry Ivanoff, born and raised here in Unalakleet, Alaska, born in 1953, so I'm 71 years old. I went to BIA grade school here in 1959, graduated County High School in 71, Stanford University in 1986. A little bit slow, but I got my degree. I'm from the native village of Unalakleet. I have a family of 12 brothers and 2 sisters. I had a family of 12 brothers and 2 sisters. I'm married to my wife Brenda for 38 years, and we have raised 3 boys and my daughter. And I'm the Bering Sea Elder Group Chairman, a group of 38 tribes along the Arctic, Yukon, and Kuskokwim River systems. And what you see up there is the map of the Arctic, Yukon, and Kuskokwim River systems, and my wife Brenda Ivanoff cutting a king salmon, which is highly prized fish we had here, but they're kind of endangered like we are. Thank you for this opportunity to make this presentation on rapidly changing systems and the verge of the Bering Sea ecosystem crash. With the Bering Sea ecosystem crash, since we're a part of the ecosystem, integral part of the ecosystem, we are a people in peril. The people of the Norton Sound, the people of the Bering Sea, it's 890,000 square miles in its entirety, the Bering Sea. Like Kristen said, I've been a hunter, a fisherman, and a gatherer my entire life. Take 4 years to get my degree from Stanford. I've been hunting for 60 years. So I don't know what kind of degree that would be, well beyond a PhD. So here's my observations. With climate change, the Arctic is on the forefront. Carbon dioxide attracted to the poles, like everybody knows. We're changing faster than the rest of the world in terms of climate. And some of the effects are Alexandria algae bloom, which leads to toxicity of our clams, and then it gets into our walrus, and then our people eat it on the islands. We're dealing with less ice coverage, so that we have concerns with plankton and krill effect. Everything's in peril, right from the phytoplankton to the bowhead, and everything in between, because we're kind of interconnected. with less ice coverage. I see krill on the river system here that was 10 feet on both sides, and I've never seen it before, just krill, all totally white, dead. As an ocean observer, top to bottom, we're having changing marine mammal migration. We're having ocean and river water warming. It has effects on fish. Without ice, we have a problem with our polar bear trying to survive. Our walrus have no more platform for their young. Our ugruk, the ice takes them around to their best feeding spots. So that we have changed the spawning rivers as warming moves north, and longer and stronger storms flooding our lower villages. Over there in Chivak in the Kuskokwim, they had the ocean right in their river, or right in their village, which they'd never seen before. One of our elders was scared for his life. Habitat changes. We have loss of coral reefs from warming and overfishing, trawling. Been trawling since 1942 with these huge boats in our ocean, and it's doing massive damage. And they're right next to the Kuskokwim River as we speak. These storms are getting more and more stronger, and it's having a lot of our villages worry about their safety. I was very impressed with the presentation by Keisha Barr on the loss of coral reefs, and that made me think of our loss of coral reefs in the Bering Sea and the effects it has on the fishing survival. With global warming, it have effects on hunting, fishing, and gathering, which we as Native people are totally dependent on. We have very little economic opportunity to get jobs to buy expensive steaks and $5.50 a gallon stove oil and gas here in Univ. So we depend on caribou, but our caribou has dropped from 500,000 to 162,000 in numbers. Our beluga, we're at the top of our level harvest of the National Marine Fisheries Service, 3%. The seals and ugruk and the bearded seals, we have less ice, as I mentioned, and the ice movement provided them a platform for feeding and for their young. In 1992, we had a chum salmon crash in northern Norton Sound. In 2015, the king salmon crashed here in Univ. I used to catch 30 in 30 minutes and a half hour. I caught 2 last year in 2 weeks. King salmon, our prize fish. The Alaska state fish is disappearing. Coho salmon, we had a coho salmon crash of 2020. I used to make $75,000 commercial fishing. My income for the last 5 years has been negative. I've lost money every year just because the fish aren't hunting. Our pink salmon in 2022, we had 200,000 come back. 7 years ago, we had 7 million pink salmon in our river system. The crab of St. Paul Island are greatly diminished. Their people are having a hard time feeding themselves now without the loss of their crab. Walrus in St. Lawrence Island and Norton Sound and Nil Diomede with no ice, the walrus are having a hard time surviving, and our people are having a harder time harvesting them. Bowhead whale, from krill to bowhead whale, like I say, with no ice, migration changing patterns. St. Lawrence Island finally got their bowhead for the first time in quite a few years. As Kristen stated, we're interconnected. We as Native people are part of the Bering Sea ecosystem. The Arctic Yukon and Kuskokwim tribes are dealing with ocean acidification, pollution, microplastics, Chernobyl and Fukushima, Bering Straits increased vessel traffic. It's the new Panama Canal, icebreakers even now to further disturb our natural habitat. We're dealing with international unrest. We're dealing with national unrest, and we're dealing with the war on education and research. And money talks, and our people never had too much money. So when you take away our food resources that we depend on, then you're putting us in a world of hurt. And the generations to come will have a hard time feeding themselves. Our ability to survive over generations of time is an oral tradition and was based on the close proximity and great abundance of everything that gave themselves to us from the Bering Sea, our river systems, and our watersheds. As stated, the Kuskokwim River is 900 miles long. The Yukon River is 1,200 miles long. There's 19 Arctic river systems from the north of Yukon to the Noatak River. They're 1,900 miles long for a total of 4,000 miles of anadromous fish streams that we as the Alaska Native tribes cannot put our fish on the table, which we've done for generations. The Kuskokwim is 48,000 square mile watershed. The Yukon is 133,000 square mile watershed. The Arctic river system greater than 75,000. So we're looking at close to 250 to 300,000 square miles of watershed produced fish. But our people cannot fish this while they catch them in with the trawlers in the Bering Sea 24/7. And then they catch them and throw them away since 1982. And the area mixed stock policy by our state of Alaska since 1960 has been catching our chum salmon and coho salmon to the tune of 1 million per year of chum salmon in Area M that don't migrate up their fish streams, and 500,000 coho per year. You look at that, that's 20 million chums and 10 million coho in 20 years. And since 1992 to the present, it's been 9,500,000 king salmon and coho salmon, chum salmon thrown away by our trawlers. You add that up, that's close to 30. You know, that's millions, 30 million fish that used to come up our river systems. And these people that are making billions of dollars are able to fish them, but while my people are going hungry. So we're begging industry, we're begging science for help. If there's any way that you can get our fish back, any way that you can protect the subsistence lifestyle of the Alaska, Arctic, Yukon, and Kuskokwim tribes, we are more than welcome to work with you and welcome any assistance that you might have. Thank you very much, and I am very happy to make this presentation, and thanks to Kristen for inviting me to this. I thought we'd do this in person, and I thought that was going to be pretty exciting, and I think it's just pretty exciting still yet for me to make this presentation, and I'd like to see a transcription of the full meeting and some, email addresses of everybody, so that if I have questions, then maybe, I know exactly where to go. Ours was an oral tradition, so I'm having a hard time with this Zoom stuff and this sending stuff and pictures and everything like that. So I asked my daughter about it, and she helped me out. So thank you very much. And open to any question if they're in there, Kristen.
Thank
you.
Oh, go ahead, Melissa.
Go ahead, Kirsten.
No, I was just going to say thank you so much, Jerry, for that valuable talk and all that information, because I really think it's a lot of stuff that most people are not thinking about. And I did receive another picture and a video that you sent. If you want me to share at any time, I can share those as well. So
those would be awesome. Those are our beluga hunting pictures where ours is an oral tradition. So in the picture you see elders and healthy cutting up the beluga and the young children in school learning about it. You got pictures of my wife cutting fish with our grandchildren learning how to do it, so there, that right there is herring fishing, which I just did yesterday on June 4th. I was out collecting kelp. I was uguk hunting on the picture to the far left, and there's a video of that uguk that just dived. Yeah, we drove right up to him. It was such a beautiful thing. God is good, and He provides us abundantly, but we're in dire straits here.
Okay.
And then the other pictures, I didn't know if we got them, was a picture of my dad butchering caribou, and then there was one where he had in 1927. There's a picture of him with absolutely no commercial clothes on. He's from Parkey, which is all made out of furs, and then his pants made out of fur, his boots made out of fur, sword made out of wood. So he had absolutely our elders have no carbon footprint. Is what that picture told me. And the change over time, rapidly changing time. My dad in 1927, no snow machines, no boats, no generators, no electricity whatsoever, so we were clean, clear, and cold. We're polluted and we're getting warmer, but we had snow today, so it's a good thing.
That's great. Yeah, I didn't get those other pictures yet, so hopefully they'll show up soon. But if not, I'll share them when they come up. But
okay, you can feel free to share them whenever they come in. But that's what it shows. It just shows pictures of the animals and our life subsistence resources that we harvest that are tied to climate change, that are tied to science. And we are the true scientists. We have never made any of our resources deplete. It was always abundant. Take only what you need and leave the rest, was what was taught to us by our elders. And
Thank you so much. There is a question for you, a couple of hands up. Jacqueline, you want to?
Hi, Jerry. It's nice to meet you. I'm Jackie. So I just wanted to tell you that there are some programs that are looking into the Bering Sea ecosystem. I did my entire dissertation on the Bering Sea, so that's my home sea in my heart. So the North Pacific Research Board has funded several different projects. I can email this to you so that you'll have a link. They have just funded some people to study the northern Bering Sea right now. And then also the Alaska Department of Fish and Game, they received funds to study the crab declines, the Bristol Bay red king crab, and then the snow crab, which have been disasters in the Bering Sea. So there are research cruises that are going to look into the causes of these and then hopefully find out, what can be done to prevent these collapses in the future. So there are things that are in the works. So I'll email you links for those.
oh, thank you so much. Any effort to preserve the resources that we depend on for life is greatly appreciated, because I don't have the money to go buy it in the store, and we don't have the stores compared to the big stores in the city. I mean,
Right.
ours are steaks that are old, that are brown in color, and $40 a piece.
And there are also
Thank you.
people who are working on looking at the harmful algal blooms. I have a project to look at harmful algal blooms with a model. And then there are people who are doing observations because I know this is becoming a real threat for the marine food resources. So.
Oh, thank you for that also, because the native village of Univ has an environmental department, and they've been noticing that the harmful algae bloom is increasing here in the Univ area, right in the town.
I'm going to email you links on all of that, but thank you for your presentation.
Thank you.
Thank you, Jacqueline. Another question for you, Jerry. We have some time. Erickson?
Hi, Jerry.
Hi, Corey.
Corey here. Good to see you. Thank you very much for representing our regions and our community and our families so well. And for those many of you don't know, Jerry has been spending just about his entire life deeply enmeshed in both the subsistence lifestyle and providing for his family. I grew up as in his house as best friends with his sons. So I kind of have the inside view here, but I have a question for you, Jerry. Being that this cohort that we're talking to today kind of represents the science world, and we definitely do recognize the huge impacts that the changing environment has, had on our community. And, I think in the subarctic, the Bering Sea region where we are, I really think that it has has had the biggest impacts, especially with the prevalence of open water during summer or winter. But being that this cohort kind of is really looking into the environmental and climate side of things, you also have mentioned the commercial impacts, the trawling that's going on, where if you guys don't know the trawling industry, it's really devastating our environment out there. So you get kind of put into the middle of the argument between as far as the fisheries die off, and specifically the salmon, you kind of get this argument between, is it climate impact changed? Is it the climate that's causing it, or is it the commercial industry that's causing it? And you being kind of in the middle of this, being kind of shot around like a ping pong ball, you got the industry blaming it 100% on climate, but then it's not very obvious that it has multiple impacts here on the fisheries die-off. How do you feel about that, where you kind of have that dichotomy of argument between one or the other? Is it climate impact changes, or is it commercial impact changes? And I know that, you and I, as fishermen and growing up in your household, we know that it is a combination, but how do you weigh those two sides of it, especially when you as a representative and advocate of our communities go into the political side where they say it's all climate caused? How do you weigh the two causes that are really have an impact on our people?
Thanks, Corey, for the question. I believe that climate has a dramatic impact on everything because of, warming. In 2019, as a commercial fisherman, I caught some salmon that were coming out of the rivers, full bellies, with milk, full bellies with eggs in them. They just died because it was too warm up the river system. That's what they said. And my nets got stinky that year for the first time. I had to cut them and throw them away. Pretty expensive. For your question, I think it's a combination of everything. You throw in climate change, you throw in ocean acidification, you throw in increased ship traffic, you throw in microplastics, you throw in Fukushima, Chernobyl, and then you got to add the commercial side of it, with the factory trawlers throwing away our salmon since 1982, and Alaska Department of Fish and Game management of our fisheries since 1962. We were the managers and we had lots coming back. We only took what we needed and left the rest. We never made billions of dollars doing it like these folks. But with the combination of the state and the federal government taking our fish for billions of dollars and all these climate things, it just multiplies the problem and exacerbates it for us as Native people trying to put food on our table. And I tell them you need to, every 10 years from 1982 to present, you need to show how many kings you catch, show how many chums you catch, show how many cohos you catch, and then next 10 years you do that. And then same way with the Alaska Department of Fish and Game. And they need to get this information together because the federal government is doing their thing, the state of Alaska is doing their thing. They don't realize that when you combine them, instead of 20 million fish that you're killing, chums, you're killing 25 million, in that 10 years. So when they combine their numbers over 10-year increments, then you can see a change in what's coming back to us. I went from 30 kings in 30 minutes to 2 kings in 2 weeks, and they're not putting no strips in my freezer. So that's what they need to do. They need to work together. And since they're the one that cause a problem, along with, God has climate change, but we have control of the overfishing. If they have any compassion for the Native people of Alaska, the United States government and the state of Alaska will work together to try to preserve the resources that we depend on. And king salmon being high in priority, our Alaska state fish, and we depend on all of our fish. So thanks, Corey. It's a combination of everything.
Thank you, Jerry, for such a nice words and wisdom that you give us today. We'll continue with this conversation and panel after we'll have another plenary talk after the break, but let's take 5 minutes. It's 3:20, and then we can come back at 3:25.
Thank you.
Welcome back. Hope you had time to have some water and stretch. Our final speaker is Dr. Richard Seavey, Professor of Oceanography at the University of Hawaii at Manoa, and a leading expert on Earth past climate systems and carbon cycle dynamics. Richard will take us back to the Paleocene-Eocene thermal maximum, a period of abrupt global warming, and his work provides a valuable paleoclimate perspective to better understand the rates and consequences of carbon-driven change and how they compare to what we are witnessing today. Let's begin, Richard.
So I'm going to talk about events in the past, not only the Paleocene-Eocene thermal maximum, the PETM, but also a few other events. But we are pretty sure that throughout the Cenozoic, that's the last 66 million years, the PETM was probably the most important event we can look at if we're interested in rapid change. Rapid in geological times is actually relatively slow because, of course, our rate is very high, so it's unprecedented. But the PETM, as I will show you, is probably the best example throughout the last 66 million years. I'm going to show a few examples of so-called events in the past. These are not all acidification events. Some of these are climate events. So you see here the last 300 million years. And here are the OAEs in the Cretaceous, anoxic events. You have some major extinctions throughout this period, like the Permo-Triassic. But you see the PETM here. So the PETM comes from the Paleocene-Eocene boundary, and there's a thermal event, so PETM. The red one here, throughout the Cenozoic, we think is the most important one to look at. There's also the deglacial transition here. This is a little bit different. It's maybe 0.15 pH units over 10,000 years. And the changes in pH and surface ocean during the deglacial transition is mostly due to internal reorganization of carbon, mostly in the ocean. So it's not a carbon input event. So what I'm going to focus on is the PETM. What's always also important to remember is that there have been large changes of pH and CO2 in the atmosphere over hundreds of millions of years in the past. So here, for instance, here are some reconstructions that we did a while ago of surface ocean pH. And as you can see, maybe during the Cretaceous or other previous times, pH in the surface ocean may have been as low as 7.6 or 7.4. But that's different because these events are established over a multi-million year timescale, and these are periods which are essentially steady states. You do not have a major perturbation at all these times, but you have a long steady state in which ocean chemistry can adjust to the boundary condition. And one argument that has been sometimes made in the past is, why do we need to worry about calcifiers? Because we know that the Cretaceous, for instance, had a very high PCO2, but here are the cliffs of Dover, and we had fantastic forams and happy calcifiers in terms of coccolithophores. The problem is, again, that the Cretaceous, for instance, is a long-term climate steady state, which has a very different chemistry. So if we go to 2,000 parts per million in the future, which I hope we will not, our total CO2 in the ocean might be in the surface 2.3 millimole per kilogram, and the saturation state will be very low, right? Because you have not, increased the weathering and changed the alkalinity. Now, the Cretaceous, for instance, might have had a PCO2 of also 2,000, but because you have had much higher alkalinity, you also had a much higher total CO2 in the ocean, and therefore a much better, much higher saturation state for calcite and aragonite. So we're looking at rates, okay? We're not looking at only low pH or high CO2. We're looking at changes, at rapid changes. And so these statements that, we don't need to worry about the future because of Cretaceous are at best misleading. So as I said, if we're looking at events, I put up here a very vague definition. Time intervals in Earth history involving large reduction in pH and saturation state on a timescale of maybe 10,000 years or shorter. If you stretch over a very long time, which I will show you for the PTM, the effects are much less dramatic because you have buffering of the system. Of course, you can talk about details. Maybe there was a few years in which the pH changed very rapidly, but we're looking for large events that actually had major changes to ocean chemistry if you want to learn something for the future. And the PTM does that. So here's a PTM. Again, Paleocene Eocene comes from the boundary between the two stages. And then we had a climate event. Some call them, hyperthermals. The PTM was hyper. Some other events were, I call them thermal. We will also see that the PTM is not isolated. For now, here's to orient yourself. Time runs into this direction. Here's the present. Here's the Cretaceous. And up here, this is oxygen isotopes from benthic forams, and gives you a scale of largely the global mean temperature, mean ocean temperature, if it was ice-free. And these dots here, this is a PTM, so superimposed on a climate that is much warmer. And the point is, you need to go back this far to have any large events that comes even close in terms of rate of what we are currently doing in terms of our carbon emissions. You can look at the Miocene, you can look at the glacial transitions. These are all interesting periods. But if you want to have a climate event that is characterized by a major carbon input and a large perturbation to the system, you have to go back 56 million years to the PTM. Okay, so because many of you are not geologists, neither am I, I'm a physicist, but just to orient yourself, this is Earth history. We know very little about this period here. This is the Cenozoic, 66 million years, that's when the dinosaurs disappeared. And here, at this boundary between the Paleocene and Eocene, that's where the PTM sits. We have, I think, now very accurate dates based on astrochronology. So this 0.01 here is not made up. I think we can do it as precisely. And so the PTM, as far as we can tell, is probably the best analog, at least for the periods and records that we have good archives for. The best analog for massive carbon release. I'm going to get to how many gigatons were released, maybe a few thousand petagrams. Surface temperature rose by 5 to 8 Kelvin for over 200,000 years. This is not a time scale that we would expect from the current perturbation. I'm going to get to this to the end because the PTM has a very long main phase, which we're still trying to explain, and we hope this is not the case for the future. We had major deep sea carbonate dissolution and ocean acidification, so you can see the acidification actually in geochemical records in the deep sea. We had mass extinctions, as you might imagine, of benthic forams, but there were no major extinctions on the continents. There were reorganizations of mammals, but it's not one of those major extinction events that wiped out, say, 90% of the species on the continents. It was mainly focused on the deep sea extinctions. Okay, so when we talk about the PTM, here's some of the modeling that we did. So you have the onset of PTM, which we call time zero in this representation. And when you look at, we have now CO2 records, we have chemistry records, we have carbon isotope records, which indicates the carbon input that was depleted in C13. And from the essentially baseline, from where you start up to the, say, highest CO2 or to the most negative carbon isotope values that we get, this portion is the onset of the event. Then you have a main phase, which this is an older paper here, which we thought was in the order of maybe 70 to 100,000 years. And we think now the main phase is more like 200,000 years. And then after this, things cool down again and slowly return to normal. And this part here, from this inflection point, we call the recovery phase. And for all of these different phases and for the different characteristics of the event, you need to think about triggers, forcings, and feedbacks, right? You can trigger deep ocean warming, for instance, by orbital forcing. You can have feedbacks that release methane from the deep sea. You can have feedbacks that release organic carbon from the continents. And you have to be very specific and careful about, you know, what are you talking about, the onset, main phase, major carbon release, and so on. And also keep in mind, the carbon release is a major cause for the carbon isotope excursion at the PTM. It is not necessarily the trigger of the initial carbon release or the climate event and the major warming increase in warming afterwards, because now you have feedbacks involved. These are all different issues. So if we look at forum records, these here are single species. Jim Zackhaus had worked a lot on this. You can follow carbon isotope values in separate species. And if you put the PE boundary, so Paleocene-Eocene boundary right here, you can look at the carbon isotope excursion. This tells you something about the carbon input. This is the same thing that's going on at the moment, right? We're pumping fossil fuels into the atmosphere, which are depleted in carbon isotopes in C13, so they're isotopically light. The same thing or a similar thing happened at the PTM. The carbon source was definitely light. So more negative values here are towards the top. And if you take roughly carbon input numbers and times that people have discussed, maybe you get 2,000 petagrams over 2,000 years is the same as gigatons. You end up with a rate of maybe 1 petagram carbon per year. If you take 5,000 petagrams over 10,000 years, you get something like 0.5 petagrams carbon per year. Now, of course, you can then compare this to the Anthropocene. And if you take the cumulative or the average rate between the 1950s and say 2000, you get something like 5 petagrams carbon per year. And 2024, if I can buy the numbers at the moment, they're about 10.2 petagrams carbon per year. So what you see is that our rates are still in the order of 10 times faster than the PTM might have been in terms of rate. But as I said, you will not find, as far as we know, any event throughout the Cenozoic, a natural event that has any rates that can come even close to these, let alone our own rates at the moment. Okay, so again, here's the PTM. So we have evidence for carbon isotopes. If you take carbon isotope record from the deep sea or from the continents, they all show a major carbon isotope excursion of about 3 per mil. So it's very common, maybe 3.5 per mil. We see in the deep sea, we see temperature changes. These are reconstructions based on different proxies. You can use oxygen isotopes in combination with magnesium, calcium in forums, for instance. You can look at terrestrial biomarkers. You can look at all sorts of temperature records. Evidence is that the average, warming on the planet was in the order of 5 to 8 Kelvin and amplitude in the high latitudes. And we have clear evidence for deep sea dissolution. So the cores, the sediment core that you can take from the deep sea, here is from the South Atlantic. And Jim Zakos and the whole crew during this expedition took course along a depth transect on the Walvis Ridge. And you will see different dissolution indicators all along this depth transect. But what we clearly have is geochemical evidence for major carbon input and major dissolution, in this case, from the bottom up, right? So you acidifying the ocean at the moment, you transfer the carbon into the deep sea, you start shoaling the aragonite and calcite saturation horizons, and then ultimately you're just going to start dissolving carbonates in the deep sea. So that's what you see here in this core. You have carbonate mostly over here, and then you have red clay where you have major dissolution, and then you return in the recovery phase to some calcium carbonate. So important is that sediment dissolution was widespread, which indicates massive carbon release. I mean, if you look at calcifiers, the problem always is it's a circular reasoning, because the question is, did they do badly because they were hit by CO2 in the surface, or were the remains that we find in sediments affected by dissolution in the deep sea? In this case, it's very clear you have major dissolution of all carbonates. That means that the CO2 was injected into or was transferred into the deep sea and then dissolved carbonates from the bottom up. But the dissolution was not homogeneous. So the CCD, the calcite compensation depth, did not rise in all ocean basins homogeneously up to the surface. What we can see is that the Atlantic was way more affected by dissolution. Here you see different sediment cores. This here is the South Atlantic. So you go from 90% carbonate up to 0% during the PTM, and then you recover all along this track here. If you look at the Southern Ocean, this is a famous site in the Weddell Sea, that's core 690 or site 690. You see there's much less dissolution across the PE boundary. And in the Pacific, there's less dissolution even in the Southern Ocean. This is a site at 2,600 meters. This is a site at 2,900 meters. So even here at 2,900 meters at the fracture zone, you see that carbonate does not drop to zero. So the dissolution in the different ocean basins was not homogeneous. We see the most dissolution in the Atlantic and less in the South Atlantic and in the Pacific. We wrote a whole paper about this. I can talk about this more later. But we can use this to infer carbon input, right? So what we do is we did some modeling. So initially we put a spike in here of 3,000 petagrams over a few thousand years, and then put it in a carbon cycle model and see how we can, whether or not we can reproduce, for instance, the carbon isotope records. And what you see in these carbon isotope records, these are the data is here in the bottom, is that the carbon isotope excursion along the main phase remains elevated, or say negative, by about 3 per mil over a long period of time, right? So this lasts what we think now more like 200,000 years. And if you want to keep we have more proxies now. We have acidification proxies, we have carbon isotopes, we have PCO2 proxies, and they all show this sort of plateau across the main phase. And you cannot get this plateau if you just start with a single carbon input in the beginning. You need sort of a continuous carbon release, some people call it bleeding, or additional slow carbon release along the event over hundreds of thousands of years probably to keep the main phase up and produce the records that we see, for instance, here in the bottom. And as I said, you can constrain the carbon input. This is the same thing, if you take antacids and you neutralize antacid in your stomach, essentially you are dissolving calcium carbonate by adding acid, and that happens in the deep sea. It happens at the moment. And you mostly end up with dissolved carbonate, calcium ions and bicarbonate ions. So you dissolve calcium carbonate by adding CO2, and that's what we see in these deep sea records at different depths. And based on this, because we know if we can constrain how much calcium carbonate we dissolve, we can constrain how much CO2 was input. And so if you have a carbon cycle model, this is relatively straightforward. You can then calculate the atmospheric CO2 change. And here we start at 1,000. So again, it was a different climate. You can start at maybe 800 ppm. You get a very similar result. And you get something like less than a doubling of CO2 with the carbon input than we can constrain, as we can constrain with a deep sea dissolution. And that, of course, then has implications for the climate sensitivity or for Earth system sensitivity. The canonical value is still 3 degrees or so per doubling of CO2. Even if you go into longer timescales, you have to think about Earth system sensitivity. And what we find at the PTM, that the Earth system sensitivity was almost certainly much higher than the canonical, say, IPCC 3 degrees per doubling, which is a fast feedback sensitivity. But clearly, the sensitivity there to climate change or for doubling of CO2 is higher than you look at the present and at fast feedbacks. Okay, so quick summary for now. There was certainly a major carbon release. It was large, but not huge. Some papers have now claimed that there were tens of thousands of gigatons. I don't think you can get away with this unless you dissolve too much carbonate. And our Earth system sensitivity from these records is probably more than 4.5 Kelvin per doubling of CO2. What I always want to point out, this is a bit more for the specialists, but the PTM is not an isolated event. Throughout the Paleocene and then mostly throughout the Eocene, you see that there are additional climate events. There are, I call them thermals, they're not hyper, they're not as big as a PTM. About 2 million years after the PTM, you had another event, which some people call the ETM2 or the ELMO, which is about half the size or less than the PTM. And after this, you get smaller and smaller events. And they're all lined up and very precisely can be dated and made on or put on a chronology with astronomical forcing. So the PTM is only the first and the biggest event that happens right at the PE boundary, but then you have more thermal events throughout the Eocene. I can talk about this later. What we think the mechanism is, but keep in mind the PTM is not an isolated event. Here is a record of benthic carbon isotopes. This is again a site from the South Atlantic. And over here, so way beyond the page, it's a PTM somewhere. And as I said, the PTM is about 56 million years over here. And then at about 54 million years, 2 million years after the PTM, you get the next major event, which is the ETM2. And then, people have now come up with all sorts of funny letters for all these smaller events here. You have what I call thermals, a whole bunch of events. And if you take the eccentricity forcing of, these are Milankovitch parameters, all of these climate events line up with orbital forcing and are characterized by a high eccentricity in Earth's orbit. So keep in mind, the PTM is not isolated. It's the biggest event and the most important one for us, but as part of the Eocene hyperthermals, and which are all orbitally forced. They all have a very similar carbon to input warming ratio. So if you just look at carbon isotope records and oxygen isotopes or magnesium calcium, you see that the ratio of these events, if you took the carbon isotopes, which are indicative for the carbon input, and if you took the oxygen or the temperature records for magnesium calcium, the ratio of these two is very similar to almost all of the events, which is indicative that the PTM also is not stands out by itself, but it's part of a series of climate events. And then if you want to have a successful theory that explains these observations, you have to dig a bit deeper. There have been lots of sensational ideas about the PTM, including a comet, for which we have no evidence, at least not that it caused a major carbon input. So these are important things to remember. Okay, so what can the PTM tell us about the future? We wrote a paper a couple of years ago where we said that the anthropogenic carbon release rate is unprecedented during the past 66 million years. And we got some inquiries from people who said that, is that a joke? Whether are we considering that the human input or humans are unprecedented on the planet? Well, of course they are. The point here is the rate, right? So if you look at any sorts of carbon release rates, I mentioned glacial, interglacial. So if you look, through the Miocene or in the Eocene, what we can tell at the moment is that throughout the last 66 million years, 66, that's when the dinosaurs disappeared. That's also a major event. There was also acidification, but it's harder to qualify or quantify. And as far as we can tell, over these 66 million years of Cenozoic, the PTM has the largest carbon release rate from natural input. And so because we are topping at the moment by probably a factor of 10, that release rate is unprecedented. And that puts us in a position of sort of, something unpredictable, because for many, at least for some of the climate changes, if you study the Holocene or if you study the glacial interglacials, you can come up with events that might be, sort of replicating parts of the climate change that we're seeing. But in terms of the carbon release and the rate, what we're doing at the moment, we do not have a good analog because the PTM was probably much slower. So what we get from these records at the onset, so remember, this is just the onset of the event. It took about 4,000 years. So at the same time, we're pumping the same amount of carbon into the system within centuries. That means that we have a no analog system, at least up to the KT boundary. I said this, then you can look at climate change. I just want to illustrate one point here, because I said again and again, the rate matters. These here are emission scenarios. So if you take the black line here, this is fossil fuel, in which we just added, 2,500 petagrams or gigatons of carbon over 500 years. So you have this sort of, logistic curve. At some point, we run out of fossil fuels, and then the emission decline. And then you can put in just a simple climate sensitivity of 3, or for PTM, you put 4.5 or so. And you can ask the question, what does this do to your climate? So here would be your surface temperature warming. This is delta T in Kelvin. And if you follow this fossil fuel scenario, you get maybe warming up to maybe 4.5 degrees. I picked a scenario here which might be sort of similar to the PTM in terms of magnitude. And the red scenario here is a PTM where we have, I think, 5,000 petagrams or so over 6,000 years. And again, the rate makes a big difference, right? What you see is that the warming commences much more slowly for the PTM. Whereas if we do this within centuries, with this scenario here, you push the system a lot faster and a lot harder. So you squeeze, the PTM essentially into a few hundred years or less. Ocean acidification, very similar scenario. In this case here, we have a PTM scenario. This is the red bar here, and we have a business as usual scenario of 5,000 petagrams over a few hundred years. I hope we're not going to do this. But if you look at the surface calcite saturation state, so they both start out at about 5 or 6 for the PTM, as well as our present scenario. And what you see for the PTM, because you're just stretching the emissions over several thousand years, you see that the impact on the surface saturation state is much less than the impact of our current fossil fuel emissions on the surface ocean saturation state, because we're doing it so fast. And essentially, the surface ocean, if you do it at a rate at what we're doing it now, the surface ocean has to take the entire burden of the carbon release. If you give it more time, you have dissolution, a little bit of dissolution from sediments, and then ultimately you have weathering of carbonates and silicate minerals from the continents that ultimately stabilize your alkalinity and DIC in the ocean. And that gives you a scenario for the PTM here, which is essentially, in terms of impact, much reduced simply because of the timescale. And that holds for calcite as well as aragonite. And so, as I said, the evidence for impact on calcifiers is much more difficult to find. There are a few papers that did find effects on calcifiers, but as I mentioned, you have issues because you have to disentangle the effects of dissolution on the seafloor relative to the impact on the calcifiers from direct input. But as far as we can tell, if anything, the impact on the calcifiers in the future is most likely to be much heavier than for the PTM because of the rate. One thing I mentioned in the beginning is the long-term duration of the PTM. So again, here you have a PTM scenario, the blue one, and you have a fossil fuel scenario where we add 5,000 petagrams over 500 years. And this is the atmospheric CO2. And at the moment, we are somewhere over here with our emissions. And if we actually were to add this amount of fossil fuel, we might end up with something like 1,800 parts per million in the atmosphere. And all of our carbon cycle models, if you take Dave Archer's model or any Richmond's models or any of these, which are now combined with fancy climate models, they predict something like the red line here. So after maybe 10,000 years or so, you would hit values of CO2 which are again closer to something where you could maybe save the Antarctic ice sheet. That's not the case for the PTM at all. So now we think that this main phase is more like 200,000 years. So the planet stayed warm for a very long time which, if that has implications for the future, means that it's much more difficult to maintain ice sheets over a long period of time, for instance. I'm not going to talk much about this. This is orbital forcing. Just to briefly say, the PTM we figured had an eccentricity value somewhere up here of 0.06 or so. And if we go into the future, you can actually calculate these orbital forcing. That's one problem that is not applicable to the future because our orbital eccentricity is currently 0.016 or so, 17, and we go into lower eccentricity. So there's the projections of having another impact from high eccentricity in the future are probably not present. Okay, so here's my summary. I said most of this, huge, but not tens of thousands of petagrams. We don't think so. I didn't talk about the boron isotopes. I can if you have more questions. And then the climate forcing, the high eccentricity is probably not applicable to the future. That's good news. So PTM relevant summary for the future. We have a no analog situation as far as we can tell because the rate and amount of carbon over a short period of time is unprecedented for the good records in the past that we have. The climate sensitivity for the PTM over a long period of time, you have to talk about Earth system sensitivity then is higher than fast feedbacks. If anything, future oceanification effects will be more severe because of the rate. Orbital forcing is not applicable, but we have to worry about the long duration. If there are feedbacks in the Earth system that can involve, deep sea hydrates, organic carbon feedbacks, terrestrial feedbacks, that could release additional carbon and even keep the climate warmer for a longer period of time than what we anticipate currently from our carbon cycle models. And I think I just stop here and take questions if I can.
Thank you so much, Richard. We have some time for questions. Amazing. Yeah, Will.
It's Richard, thank you so much. If you can briefly explain why different portions of the deep ocean had very different sedimentological response to this massive event. How can that be? What was so different about deep water circulation, or why do we see what we see, simply speaking?
Yeah, so it has to do with ocean geometry. So, during the PTM, the Atlantic was much smaller. So it was like a tiny ocean basin. And before the PTM, you had mostly deep water formation that in the Southern Ocean, which means that the water mostly spread from the Southern Ocean into the deep Atlantic. Now you dump in carbon, and that's where initially it first goes. It is sort of concentrated in a small basin. And then ultimately it gets transported into a larger basin like the Pacific. And relative to the area, you just see a muted response of the CCD. Another factor is that, some people have evidence that probably if it was, feedback through methane hydrates, that those were, predominantly concentrated in the deep Atlantic, which would, add to the carbon release in the deep Atlantic. That's why we see the big difference between Atlantic and Pacific.
Yeah, from Norm Nelson. Thanks, Richard. That was very informative. What is the current thinking about what comprised the initial large carbon input in the PETM and then the sustained lower release? What is fossil carbon from an earlier epoch?
Oh, so again, we have to differentiate between triggers and the carbon input during the main phase of the release. And so if you think about triggers, that could be a lot of things. We favor orbital forcing, for instance, because we know that the hypothermals are triggered by this. You could have like a small input of volcanic carbon in the beginning. You can also have just internal reorganizations. That's a paper that goes back to the 90s. Of an internal feedback in the climate system that just keeps, starts warming up the deep ocean. And then you can release methane hydrates, for instance. So our favorite sources of carbon for the event are deep sea hydrates and organic carbon, because they have carbon isotopic compositions that are consistent with a moderate carbon release. Some other groups have favored volcanic carbon. I think that's a problem because volcanic carbon has an isotopic composition of carbon of -5 or so, or 6. And you need to dump a huge amount of carbon into the system to get the carbon isotope dispersion. At the same time, when you do this, you release, you dissolve too much deep sea carbonates. So, whatever the carbon fossil portion were at the time, methane hydrates, right, are stored over a long period of time, over millions of years in the deep sea. If it's organic carbon, it's carbon from the continent, which is essentially comparable to our fossil fuels. So these are the sources that are seriously being discussed.
I have a question. When you talk about recovery time, you say like conditions that were previously than the event, or is there another recovery? How do you define recovery time?
Yeah, so essentially when you look at the carbon isotopes, essentially they show the excursion and you have a main phase, and when they start declining and CO2 starts declining, you have a very long tail over which the system goes close to back where it was before. Not exactly, because you're sitting on a long-term trend over millions of years, but you can define the point at which you essentially have recovered to, say, 90% of the initial perturbation. And that takes another 100,000 years. So the tail, even for the recovery, is very long.
Thank you. We can open for the panel discussion.
I have a question for Zach. I want to know, what is the mesh size of the samples you're working with? If it's just the 500 micron bongo toes or the 200 micron propose, and if you're also like thinking of making some kind of time series gridded product to be available to the scientific community.
Yeah, great question, Colleen. This is for the this first round is for the bongo net toes. So the 505 micron. So they go from about 210 meters to the surface. My understanding is that the Propos vertical net toes are all preserved in formalin. Separate effort that we're trying in our lab is there's been some recent developments to get DNA from formalin that have come out of Japan in the last few years. And so we're playing around with that this summer, where we have some zooplankton toes that have already been counted in formalin. So we know what we should be able to recover in the jar. But stay tuned if that is possible. Unfortunately, I do think that sampling is destructive. And so it'll be probably a longer time out, and we have to be more thoughtful about, how we might decide to generate DNA from that. Whereas in this method that we're using with the eDNA, we don't hurt any of the actual samples or organisms, which is nice in that, it was easier to get permission to be able to put ethanol off the top without destroying it. And to your gridded, yes, that is the plan. And so the goal is to put it into an ERDAP. And so we are working on the behind the scenes to be able to handle how do we actually put genetic type data and merging fair like eDNA standards with standard NOAA ERDAP serving tools. And then the nice thing about having an ERDAP is you can get it in NetCDF or any kind of other product, CSV, tabular, you name it, that you might be interested in.
Are you detecting plankton that wouldn't be caught in that net, like smaller zooplankton or the phytoplankton that those zooplankton would have been eating?
We are not getting phytoplankton, not very much at all. And there are definitely some smaller zooplankton that we might not expect to get caught in that net. And it is a question of how did they end up there, whether they were in the stomachs of one of the organisms that were already, there, and then that's what we're sampling, or if there is high enough biomass that the functional net size actually declines over time to be like a smaller mesh size. I know that's more of a problem for eDNA filters that we've been thinking about, but I'm not sure if that happens too for those kinds of nets. But anyway,
not that much. It'd have to be a really full net.
Yeah, I would imagine so. Some of them are pretty full. Pretty full, but not that full. So.
Richard, thinking about, we heard this week a couple of estimates on marine carbon removal and ocean alkalinity enhancement. And I was just thinking if you have ideas on how much calcium carbonate will have to dissolve to neutralize these carbon inputs and the period of time that will actually be needed.
This is essentially a mole per mole basis. So you can actually look up some, we published on this, David Archer published on this. Ultimately, as you transfer the CO2 in the deep sea, you dissolve the deep sea carbonates. And this will, but it will take tens of thousands of years until you actually start doing this. But, you know, basically it's roughly on a mole per mole basis.
Rachel, do we have some questions?
We do have questions. Oh, but I see Jerry raised his hand.
Yes.
After Jerry, I'm happy to put in some of our questions.
Yeah, hi. My question is regarding carbon release. Not only are we attracting so much and causing so much problem with climate change, but with the warming of our climate, we're having a lot of permafrost melt, which is releasing even more carbon, you know, that over time. So with the change in climate, we're also seeing an increase in the carbon release by the permafrost melt. We're having deep sinkholes here, even around the Unifleet area. I know 3 sinkholes. So I know that the permafrost is melting, and from what science I'm able to read, that releases quite a bit of carbon into the atmosphere also.
Richard, did that happen during the PETM too, the permafrost melting, do you think?
Yeah, it's a good point about feedbacks, right? I mentioned feedbacks, and once you start the warming, that's probably similar scenarios at PETM as in the present. The permafrost is a big issue. I talked about deep sea hydrates during the PETM. People have actually looked at this, and the situation currently for the deep sea hydrates is different than for the PETM. We may not expect a major release for carbon from the deep sea. Permafrost in the Arctic, for instance, is clearly an issue. Estimates are in the order of maybe a few hundred gigatons. So that's not good, but it's not thousands of gigatons. And there was actually one paper on the PETM which claimed it was a major release of carbon, permafrost carbon release from the Antarctic. I'm very skeptical about this, but yeah, these are important feedbacks, no question.
Another problem that we have with the release of that carbon is, or the permafrost melting, is methane gas. There's a lake up there in Kotzebue that has methane gas coming out like you wouldn't believe. I mean, you know, they cut a hole in it and then they light it with a lighter and it actually burns up 6 feet into the air. You know, so that's quite a bit of methane gas that's being released, you know, by the permafrost melting, and it's a steady flow.
Oh yeah, no doubt. You can dig up methane hydrates from the seafloor and light them up. They burn. But of course, you have to look at the total emissions relative to our fossil fuel emissions. And methane is not a good thing. But you know, if people talk about strategies about reducing methane or carbon dioxide, methane has a short tail. It's not good. It's a greenhouse gas, no question. But it's a relatively short lifetime. If you want to do long term, you should focus on CO2.
I think for the sea star decline as observed in the eDNA, is there any hope, belief that these populations may reestablish and that the eDNA may be able to detect recovery, perhaps more relevant for other taxa than having been all but eliminated?
Yeah, I mean, there are still sunflower stars kicking. And actually, California just saw its first stars since after the sea star wasting disease hit. So one of them is named Brittany because apparently they're naming them all after pop stars, which is adorable. And so there is hope. One of the things that has been, you know, maybe giving a lot of people a ton of hope is in the last 4 years, we had never aquaculture grown a single sea star. And now we figured it out and completed the entire life cycle, which is really incredible because anyone who's ever dealt with aquaculture, like keeping planktonic larvae alive and having them settle successfully, is why we do not have a lot of aquaculture industries for a lot of species. So there is hope, and the Association of Zoos has their safe program, and Pycnopodia is a species in the spotlight, and there are at least 30 aquaria across North America now that are actually starting a captive breeding program, including some with individuals from California and Oregon from, that were in basically aquaria before the sea star wasting disease hit. So there are some unique, like stock, questions. There's active work looking at population genetic structure. And they're certainly gone in Oregon and California for the most part, certainly functionally extinct. But there are parts of British Columbia and even Friday Harbor Lab where I've gone diving to see them, where there's similar abundances as before the marine heat wave. So I think there's definitely hope. The species is doing well, particularly in colder climes. And yeah, there's definitely a lot of unknowns about, the success of recovery. I think, don't care how good of an aquaculture program you have, that's not going to, get 5.5 billion individuals back into the environment. But there definitely are active like restoration efforts. And that's where we think the eDNA twofold might be really helpful. One, as you can imagine, I mean, they are disappeared just along the U.S. West Coast is, 2,600 kilometers of coastline. There's no way you're putting divers, in the whole, coast to be able to identify where the last refugia are. But you could theoretically take water samples from Niskins on, on boats and do synoptic surveys. So this summer, we're planning on doing basically a rapid field assessment of hopefully somewhere between 20 and 50 sites in Northern California to identify where some of those last individuals might be. And then, on the flip side, doing the recovery efforts, as much I'm a scuba diver, I love diving, getting underwater is amazing, but it's very time and labor intensive. And so being able to, track recovery without having to, put divers in the water every time will be super useful. And so that's where I can see the eDNA being really helpful. And particularly because Pycnopodia are known to go all the way down to, 400 meters depth, right? And so there's no way anyone's scuba diving down there. And so it might be really helpful to look at potentially some of those deep water refugia. That's like a sort of big unknown question about the species right now. Obviously, visually, no one's seeing them. There's no reports of seeing them in a lot of the coasts where, we recreate. And it's really been recreational or the commercial fishermen that have been, the other one that is not named yet was found by a commercial urchin diver. So those, the people who are in the water every day, for a living livelihood, those are the people that are finding them. And so, but there's a big question of, all right, well, what about the places where no one is viewing them? Are they safer at deeper waters? And we know that some species have, been able to, go deeper depths to dodge some of the warming, if at all possible. But I will say, sort of relatedly, kind of just like Richard's whole talk got me really freaked out about how fast we're changing the biogeochemistry of our systems, and so much faster than anything on record. And, obviously a huge carbon bomb. And what does that mean for our species and ability to, adapt to these really rapid changes that don't have a potentially evolutionary analog? Definitely is going to keep me up at night for the next couple of weeks.
Zach? Yes, hi. I realized, so this question is for Jerry, and it's actually very similar to one that Patricia just put in the chat. Yeah. But I wanted to ask because Heather asked in the chat about, any tips that you might have for scientists who are interested in kind of collaborating with Indigenous tribes. And I was interested if you had any examples maybe of ways in which you've seen this go especially well. I know you mentioned in your response some things about clear lines of communication and access to data, but I was wondering if you had any kind of just examples you could give of where things really went well or where things really didn't go well. And this actually might kind of align with Patricia's question too about kind of sharing stories about ways in which conservation worked really well in your tribe.
Thank you, Zach.
In terms of conservation, in the tribe, it's always been take only what you need and leave the rest. And there was no commercialization of any of our products, any of our resources. So money was not a part of it. So it was not volume that was taken out. It was only taken enough for us to make the next season. Every year, each cycle, fall, winter, spring, summer, provides us something different to put on the table. And over time, that's changing. And I'm just really worried with the climate change and everything that's happening in terms of management, that we'll have very little chance to get our salmon back if nobody's listening. If we continue going after the billions of dollars, then our resources, our salmon resources, are in trouble. In terms of warming climate, I'm not too sure what we can do about that. There's greater minds at the Paris climate summit, that know what's happening. But what I'm seeing is just things that are happening to our resources because of the management and because of climate change. And how can we make it better? I think through communication. I've testified before the Fish and ADF&G board for 80 years, or since 1980s, and I never won one. The board has 7-member board, and the mixed stock policy folks know that it takes 4 votes to win their way. So they always have those 4 votes already in their pocket. I don't have a war chest to go wine and dine these folks, so I watch them as they're wined and dined, and then they vote that particular way. And the same thing, I believe, with industry, with the North Pacific Fisheries Management Council. As long as they see those billions of dollars coming in, they are happy. The United States is doing good, but effects that it's happening to our tribes is phenomenal. It's drastic, and it's putting our people in grave danger. We're a people in peril. And if we've got any research that can help us along the way, whether it's all the way from, like I said, from krill to bowhead whale, and everything in between is in danger. So we need to protect all species, not only the krill and the zooplankton. I never thought that those would be important things, but after I saw 10 feet of krill on either side of these creeks, because of a huge storm, that's never seen before in my life. So communication is key. And if there's any way that industry or science can help our tribes in Alaska to continue our lifestyle, we welcome any kind of information, any kind of knowledge, and be glad to work with anybody that wants to work with us. And Bering Sea Elders Group, out of, like I said, 38 tribes, and we do have a Facebook page, and all that information can be found there. And if there needs to be any correspondence, then with the elders, I do believe there was a beringseaelders.org email address there too. Communication is key. Right now, we're seeing a lot of information being passed to and fro, but we need to be heard. The people, the scientists, the managers need to hear the plight of the Alaskan tribes, because it's having a real tremendous impact. When you breach that oral tradition that's not written down, if my kids and grandkids don't get king salmon, then they're not going to learn how to make king salmon strips. So one of their resources that we put on the table is lost forever. So it's scary. It's scary to me. So yeah, any help that we can get from science, from managers, from the state, from the federal government, from the international governments, because we got them killing our fish in the donut hole beyond 200 miles. I was thankful for that 200-mile limit when they passed it, but trawlers, American trawlers came in and they did the same thing. They're doing the same thing that international trawlers are doing. So whether it's their governments, international governments doing it, or our trawlers doing it, the damage is still there. And then you add climate change to it, carbon release and methane gas, and it's the Bering Straits as Seward, Panama Canal. It's easier and more efficient to go and save money, but then that endangers our resources too. Oh, thanks for the question there, Zach.
Thank
Hope
you.
I
Thank you, Jerry. And I think that wraps our section today. And this is one of the, we wanted to bring Jerry here to pass that oral knowledge to all of us. And I don't know, Kirsten, you have a couple of thoughts to close our session and then pass it to Heather.
Yeah, just quickly, I just think that this was a great session spanning so many different topics of rapid changes in the ocean. And yeah, I'm kind of just processing all the different information that I heard from, the coral reefs to Jerry's stories about the Bering Sea ecosystem crash. That's like totally scary. And, Zach's fish and zooplankton stories, which are super interesting, but also offer a little bit of hope, especially for, seeing changes in the future of what's going to happen. But yeah, it's been a really interesting session. So thank you to all of our plenary speakers that spoke at the session. And thanks to Heather and May for helping us organize this, especially the pivot to a virtual format, which was super helpful. And I think it went well. Okay. All right. Thanks.
Thank you for that fantastic session to wrap things up. I mean, talk about covering a broad range of timescales and species, different aspects of the food web, as well as all over the globe. You guys went all over the globe. It was great. Thanks for that. Thank you to our speakers for the entire OCB workshop this week. I'm going to just share some final remarks. They will be quick, and then I will let you all get back to your weekends, which should be starting soon. Just want to say thank you to a lot of people. Our first and foremost, the NSF and NASA program managers who have been and continue to support OCB in every way that they can. And Woods Hole Oceanographic, which is where it's our institutional home. OCB is global, but our little office is here in Woods Hole with lots of plants and plenty of coffee and tea. So if you ever find yourself at Woods Hole Oceanographic, we'd love for you to visit us. I want to point out, we usually get, when we have in-person meetings, we usually get support from NOAA GOMO. And although NOAA doesn't support the project office, they support an enormous number of field and monitoring programs and a lot of things that this community relies on heavily. And you saw some of those this week when we talked about biogeochemical and talked about the East and West Coast ocean and actually Gulf of Mexico and Alaska coastal ocean acidification monitoring programs. I want to thank the Scientific Steering Committee and the session chairs. I've been going to multiple biweekly meetings, figure for 4 sessions. That's quite a few meetings biweekly meetings over several months. We've been planning and plotting this since fall. And it all starts with you. We ask for your input on sessions, both at the end of one summer workshop before the next one, but we also put out a call in the fall and say, hey, send us your ideas, because in the fall we make decisions about which plenary sessions are going to go for the following year. So keep doing that, because we listen. I think 3 of the 4 sessions this year were started as proposals from a member of the community. And then the SSC will work with that community member to plan the sessions. I really want to thank all of the plenary speakers for not only presenting amazing science, but for presenting it in a way that it was accessible across the range of disciplines and roles that attend an OCB workshop. You brought your A game, and we really appreciate that. This workshop is all about spanning those big green icons that I showed at the beginning. It needs to click across disciplines, and y'all did a great job. So thank you for making our meeting so stimulating and fun. I want to thank my team, May and Mary. You all know them well. If you haven't met them, I hope that you meet them in person someday. Most of all, I want to thank you, because it's the people who come that really make the meeting, and seeing all your faces every day online has been wonderful. We could not have imagined how much participation we were going to see online, as we've seen this week. I appreciate your grace and your patience with our last minute pivot to virtual. We did the best we could. Over the week, we had almost 500 unique visitors. That's just from Monday to Thursday. That doesn't count today's session. We won't have those data until tonight, but we pretty much sustained around in the neighborhood of 250 unique visitors every day. So that's a pretty good turnout for all of you. So kudos to all of you. Again, there are plenty of ways to get involved with OCB. I already shared this slide, but if you want to get on a Zoom call and I can kind of walk you through some stuff or point you to some stuff based on your research interests. I've been with this program for a long time and have a lot of institutional knowledge and gray hair to go with it. But it's been a real honor and privilege to work with all of you through time. And we've done a lot of great things together, and I hope to continue that with you. Finally, as many of you remember from last year, we had just completed an external decadal review of OCB. And one of the big outcomes of that, there were several recommendations, and we incorporated much of the input we got from you and from that review process into the renewal proposals that went to NSF and NASA last summer. But one of the big pieces of advice was on strategic planning. We need to stop and take stock of what we're doing and where we've been, and that really helps inform where we're going. And so we are a bottom-up grassroots operation, and we really want to hear from you. OCB is very different from most other programs. We don't have a science plan. It's not how we operate. We have those green icons, those pillars, and that really puts the guardrails on our program. We have research areas listed on our website. They're continually evolving. As I talked about throughout the workshop, we've risen to challenges like marine carbon dioxide removal discussion, or extreme events, and thinking about how we can organize and coordinate this community better to be able to be more responsive. How can we leverage resources that we already have, technology that we already have? So I encourage you to spend 5 minutes, take this survey. It is a survey both about the workshop, but also about OCB's future. We want to hear from you about how we can best support you, especially over the next couple of years. We hope to be at Ocean Sciences in Glasgow in 2026. OCB will be turning 20 at that point. So we hope to see many of you there. We're also going to send this survey link out to the email list. So if you don't want to take it now, you can take it later, but please take it. It really won't take you that long, and your feedback really is helpful to us. And that's all I have. Thank you everybody so much for your attention, for your presence this week. It's been a lot of fun. It's been great to see you all.